Phones off. Everybody has cell phone off.
That was Kelly, our documentarian, with perfect timing. Are you on?
Of course I'm on.
Okay.
I gotta get the mayhem of you going through this.
Welcome to Summit THRiiVE number 13. This, my dear friends, is Dr. Christopher Shade, and I like the way he looks better than me, so I'm going to exit. I was going to do a whole spiel at the beginning, but instead I'm going to say that following Summit number 12, okay, fine, I recognized that one thing we really needed to focus on was redox. And if you don't know what redox means, by the end of today, we're hoping that you will. Christopher Shade is nothing if he's not a pragmatic. Is there like a brilliantist? Is there like a noun with the word brilliant in it, or just brilliant? Okay, well, we'll go with that.
What am I to say?
And I'm starting with him because at Autism One, he presented for me at like 8:30 after everyone had cocktails. And he's not only brilliant, he's hysterical. And he really wants you to understand the science to be able to make wise choices about all this stuff. And then the name of your company is QuicksilverScientific.com. And he makes amazing products from what I've heard. And we will start with Dr. Christopher Shade, who's a PhD, by the way. And oh, Robert, so glad you could make it.
Wow.
Okay, Christopher Shade.
Sorry, I didn't bring the walnuts in time.
No, you didn't. You can make no noise and come on in.
Thank you, Dana, for bringing me in here. Thanks, everybody, for showing up. I like situations like this where there's not a lot of people, and it's really just thought leaders where we can talk about all the things that are going on in integrative and functional medicine and try to get to some consensus on things. And one of the biggest things that needs some consensus right now is antioxidant therapies versus pro-oxidant therapies versus things that we think are antioxidant therapies that are actually pro-oxidant therapies. And why would they both work, and why would they both do actually the same thing, and what are the subtleties? That's what I hope to bring. I'm going to talk first.
Wait, Christopher, I have a question.
What's your question, Dana Gorman?
Oh, Sarah Corshane, thank you so much. We're just wanting to make sure people know that at this summit, we have a system for when people have questions so that they can hear them as well as what you're saying. Thank you.
Okay, so if anybody asks me a question, I have to tell them to take the mic?
No, you just have to pause. She's going to get the mic.
Ms. Corshane will travel with Mike.
Exactly, and the people out there in Google land are thrilled right now.
Hello, Google land. Okay, I'm going to run through a bunch of stuff today. I'm probably going to take more time than Dana wants me to take, but
okay.
She'll be happy with it as we go through there. We're going to talk a lot through the day about radicals, like ourselves, free radicals, reactive oxygen species. Everybody talks about ROS. They don't talk about RNS, reactive nitrogen species. I'm sure Gary knows that this is where everything disperses into. Reactive sulfur species. How are all these things the same? How are they different? We'll talk. I pulled a lot of stuff out of this book. I brought this book if you want to peruse it. It's a little nutrient-dense redox signaling and regulation in biology and medicine. But it really sheds a lot of light on what we're going to be talking about. And using my very fancy iPhone scanner, I've taken a bunch of stuff out of there. So each slide isn't really referenced. If there's not a reference on it, it's probably out of this book here. So after we look at this first slide, I'm going to drop into my framework of what detoxification means and what the core of detoxification and the different proteins and enzymes involved in that, because they're at the core of the problems that we're seeing in health. They're at the core of why certain proteins aren't functioning, why certain genes aren't firing. And our goal here is to clean those things up and get them back online and make them work again. And so we're going to talk a lot about how free radicals fit into this. And I was very glad that a lot of things have come along, like ASEA, like ozone therapy. Peroxide therapy has been around there a lot, but it hasn't come to the fore like some of these others have, because they forced me to look back into how I was using plant-based therapies, and we would call them plant-based antioxidants. And what I found really is that these things that we think are plant-based antioxidants are actually have a very strong pro-oxidant character that create free radical reactions that enable things to happen. So one of the biggest areas, this is pretty technical talk because I get really annoyed at the language that's used around the healing world and how everything gets put into a metaphor and how we chase down various things like, is this chlorine dioxide or is it chloride? Well, really, a lot of these things that we're using as pro-oxidants distribute into a lot of different pools in the body and become a lot of different. This is a map of your main reactive free species or radicals, and they have them color-coded so that in green they're totally non-harmful, and as you get farther into red you get more and more harmful till you get to the peroxyl radical, which is no doubt a damaging thing. And then you have a lot of these in-between molecules that can go a lot of different ways, like hydrogen peroxide is a core one. And as it reacts with things, it goes into different pools. So it's very hard for us to say that when we take a pro-oxidant that we're taking this one specific thing. You know, maybe when we inhale from a Valkion, we've got some pretty clean singlet oxygen, but maybe we don't. And once it reacts in the body, it becomes all these different things. When we do ozone therapy, I mean, how long does it take for ozone to react with all the blood lipids and create all of these oxidant lipids? You know, it's like fractions of a second. So it's really these ozonides, these products of oxidative reaction, are doing the jobs in the system. So we'll go back to that a little bit longer, but this is, you're going to talk more about differentiation of the different reactive oxygen species?
Yeah, I actually have charts on it.
Okay, good, good. Oops, wrong computer. Let's try this one. So I'll run through a little bit of the detox system and understand, which is mostly an antioxidant system. It's the response of your antioxidant system to things that's taking metals out of the body. And then we'll look at how it works, how it breaks down and doesn't work. We're going to look at this super system, this thing I call the antioxidant detoxification protein repair super system, which is a group of enzymes and transport proteins that all work together. And actually, these other two we're not really going to talk about. Just on the detox, when you're looking at metal resistance, you're looking at mercury, cadmium, and arsenic, there's one central set of molecules that give you defense to that. It's glutathione. It's glutathione as transferase, which is a phase 2 enzyme responsible for catalyzing the movement of the metal off of the cellular protein onto the glutathione. And then you've got transport proteins that take it out. And this is just an abstract from a paper where they were looking at cell cultures. They made all these different cells. They kind of take cells and they bombard them with gamma rays till they create mutations. And they find like a super cell line that can swim in metals. And then they see, well, what has it got in it? And those are the things it had. It had a lot of glutathione biosynthesis, had a lot of glutathione as transferase, and it had the transport proteins. So you have those in line, you're able to get rid of the metals, and you're able to get rid of a lot of other xenobiotics or toxins that are coming into your system. So we need all those things to work, and there it is just laid out. Glutathione sufficiency, glutathione as transferase, and the transport proteins that are clearing things out. So so we're all on the same page, I'm just going to run through these. Phase 1, people used to talk about phase 1 and phase 2 detoxification, and they generally missed phase 3 because there was a lot written by Jeff Bland and functional medicine before the year 2000 when they discovered the transport proteins. So phase 1 is oxidative activation generally creating a free radical, moving it into phase 2, which is conjugation, then taking it to phase 3, which is transport out of the cells. So phase 1 we associate with the P450 system, cytochrome P450, and it's making free radicals out of something. So it's really important that it moves into phase 2. In fact, in the story of breast cancer, they find that there's a lot of estrogen-free radical created in phase 1 but not moved into phase 2, and that estrogen-free radical is abstracting base pairs off the DNA, and they find women with breast cancer or breast cancer risk factors being a lot of peeing out a lot of estrogen DNA adducts, meaning that they have this phase 1 reaction going on without moving it into phase 2, which would be conjugation to glutathione and moving it out of the system. So it's very important to bring those phase 1 and 2 together. Now for metals, metals are already really free radicals, and so they don't need this step. Phase 2, we're going to make things soluble, water soluble, and recognizable by the transport proteins. These, we were talking about this last night, these conjugates from phase 2 are anionic conjugates. They've got a negative charge which keeps them water soluble, and they move through the transport proteins. When we talked last night about binders, you look at something like polystyramine, it's an anion exchange resin, and it's able to grab the conjugates that come through in the bile and hold them so you don't reabsorb them. In the natural world, chitosan, which is derived from crab shells, has a natural anionic character to it, so it's the natural version of polystyramine. And of course, we see either functional or genetically low expression of phase 2 in people that accumulate a lot of mercury and other heavy metals and toxins in the body. You'll also find when we get deeper into this, so we're going to look at broader layers and then get deeper into what's the dysfunction that stops these things from happening. A lot of this is epigenetic dysfunction. They used to suspect that people with prostate cancer had GST problems. They couldn't make enough, they weren't using glutathione transferase right. But then when they genotyped everybody, they couldn't find any difference between the cancer guys and the non-cancer guys. A little bit, some did, some didn't. But then when they went into the tumor itself, they found that glutathione transferase promoter genes were hypermethylated, meaning they were turned off. There was no expression of glutathione transferase in the tumor, which now brings us back to Dietrich Klinghart's long them looking at tumors and finding a lot of toxins in the tumor. So there was the take-home message was that the tumor was creating the toxin was creating the tumor. But now we look at it another way. The tumor is creating a dysfunction which stops detoxification, which then accumulates toxins. It's also stopping other reactions, which are anti-neoplastic reactions. So this accumulation of toxins, is this a secondary effect or a primary effect? So this is a lot of what I want to do is go into what we've seen. I mean, we see that these therapies do things, but if we don't know the correct mechanism, we can't advance to the next stage of how to bring our therapies forward. And then phase 3 is to transport out. So this is super important. You've got, I mean, how many different phase 1 reactions? I mean, there's so many different phase 1 reactions. You know, you've got dozens of those funneling down to a handful of phase 2 reactions, funneling down to really a single transport system that services all of it. So it's an inverse pyramid coming down. So when the transport system is down, you're blocking a lot of things. You're blocking glutathione conjugates, glucuronic acid conjugates, sulfate conjugates. And of course, you have the highest number of these transporters in the liver, then the intestines, then the kidneys, right? Because you're bringing stuff out. So to look at it schematically, up in the cell here, we've got phase 1 drawing down into phase 2. Then we've got our cellular transport protein. These are transmembrane transport proteins that use magnesium and ATP. Hence why one of the reasons magnesium is so important in detoxification, and one of the reasons that it takes a lot of energy. You're consuming ATP to do this. You bring it from the cell to the blood. From the blood, it's going either through a transport protein into the hepatocytes, and then another transport protein that dumps it into the small intestine, and then we move off to the sewage treatment plant. Some of these transport proteins are located right along the intestine, and then of course the kidney has got them as well in the tubules. So this is not a glomerular filtration process. This is active transport into the tubules. Just, you know, Dana likes, you know, non-engineer-like slides, and this is as happy looking as I can get. At least I have a cell. So you've got phase 1 and 2 in the cell. You've got transport out of the cell. And of course, before I said right to the blood, but we know nothing goes right to the blood. Nothing comes right from the blood. It all goes through the extracellular matrix. So the matrix is that place in which conditions are made or broken, for nutrient transport into the cells and detoxification out of the cells.
Chris.
We know that that's where all the energetic work happens.
So you're talking about these transport proteins being in the cell membrane?
Yeah.
So when somebody has a detox reaction or a P-tox reaction, and you're talking about these terms that we use, what does that mean to you?
Give me like 3 more slides.
Okay.
Cool.
Yeah.
Perfect.
Yeah, because there's a point at which this is going to come up. So this is how everything's supposed to work. So you've got stuff dumped from the cells through the matrix into the blood, and then it's working its way through the kidneys or the liver into the small intestine. And then you've got all these things. Remember, they used to talk about phase 1 and 2 as phases of hepatic detoxification, as if it was only happening in the liver. Well, what's the rest of our body going to do? You've got this in all the cells, but maybe you have 1 or 2 copies of the chemistry in the cells, and you've got 30 or 40 copies in the liver, because that's what they do. So they're taking first pass circulation, detoxifying a lot, and getting rid of it. But you've got to do that everywhere. Otherwise, how are you going to get rid of the toxins in your thyroid? How are you going to get rid of the toxins in your brain? You have them everywhere. So how do we disrupt that? So that was, we saw how it worked normally. Now, one of the best ways to disrupt all of this chemistry is inflammation. So inflammation in the small intestine dramatically turns down the activity of this last transport protein. Once you turn that down, you have a negative feedback back into the whole system, and you turn down everything. This is all a conveyor belt going out. And if you can't go out, the signals cascade up the line to turn everything off. Then you have this buildup of cellular and bloodborne toxicity, and you're holding everything in. Now, the one thing that doesn't happen is turning off phase 1. So phase 2 and phase 3 are probably evolutionarily linked so that they go up and down together, but there doesn't seem to be a good feedback mechanism into phase 1 to turn it down. So phase 1 keeps creating free radicals. Remember, we create the estrogen free radicals where we have a lot of estrogen, and then we have the mutations happen. And so here you've got GI inflammation, toxin retention, and internal free radical generation. Not the good free radical generation, the bad free radical generation. So that's a picture of chronic disease. It's a great picture of autism. And, you know, this is just showing you data where they wanted to look at how these transport proteins and these different groups of detox elements come up or down with inflammation. So how do they induce the inflammation? They use lipopolysaccharide. So that's the cell membranes of gram-negative bacteria like you would have in infections. And what do you get when you get leaky gut? You get lipopolysaccharide translocating into circulation. What do you get when you've got real bad cavitations, root canals, sinus infections? You have a lot of lipopolysaccharide moving into circulation, and it creates inflammation. And what does that inflammation do? Well, this is one of the main detox gene sets, and you see it turn down 60%, goes to 40% of its normal activity across the whole GI tract. And then these are subsets of these different proteins inside the hepatocytes, inside the liver. Down, MRP2 here is the route through the bile to the GI tract. That gets turned way down. What doesn't turn down is MRP3. This is a door from the liver back into circulation so that you can remove things through your kidney. So what that's saying is when we block, usually you've got liver kidney routes, you block the liver GI route, and then the load gets transferred over to the kidney, and you increase the kidney load. Now, the kidney, so here's P-tox. You start jamming the kidney because you blocked the GI and liver. And it's mostly, I think, liver. You blew out the liver, then you go into P-tox until you trash the kidneys. Because there's no better way to trash the kidneys than lipopolysaccharide. Actually, than lipopolysaccharide and metals. So I talked for a long time about retention. I mean, everybody's talked about retention. You know, Hal Huggins wrote about it in '73. They got all these amalgams, but there's no mercury in their peat. And our testing shows it real nice, but I didn't have a great model for how it's done. And they got mice and they injected them with mercury or lipopolysaccharide, and it didn't affect the kidneys that bad. And then when they combined the two, wrecked the kidneys, serum urea nitrogen went up, bodily mercury went up, and urinary mercury went way down. So you first blow out this, you translocate over there, you blow out that, then everything's blown out.
Can you slow that
Wait.
one step back? So when you say you blew out the liver or you blew out the GI tract, can you just repeat what
Okay.
you
And we don't like words like blowout and trash. We like words like challenge and,
Well, the word is fine.
Why do we not like blowout?
well, because these parents are struggling, and when you say words like trash, the parents sitting there
They
going,
feel like I just trashed my kids' kidneys?
no, they feel like, oh my God, I've been pushing like crazy on this protocol. I've had P-Tox for 8 months, and this
I
guy
just
just
love
said I trashed my kidneys. Holy shit. This.
Okay, all right, wait, let's go back.
I'm
Okay,
with the
now we
term
challenge.
here. But I know you went through the different transport proteins. Is there a specific transport protein that you're talking
So
about?
there's a transport protein going from, so these are all these phase 3 transporters, right? And there's one that goes from the hepatocytes, from the liver cells, draining through the gallbladder into the GI tract. And that's the one that gets turned off when you've got inflammation. When that happens then, the transport protein, the MRP3, stays open, things are forced back into circulation, hoping to go to the kidneys, and then the kidneys pick up the slack. This is why you'll see, if you work on metals in the GI tract or inflammation in the GI tract, people who had to wake up a lot and pee many times a night, a lot of that goes away. There was a, older naturopaths, you say, would tell me, oh yeah, we used to say if the kidneys are shot, look to the GI tract. And that's for this reason.
So the transport protein between the liver and the gallbladder that dumps into the intestines is blocked.
Yeah.
So it has to use a different route.
Yeah. And it's the same one that is the bile transporter.
Yeah, so
Do you
from
want me
now
to, or
on,
you?
the responsibility is to either have a mic or the person presenting has to repeat
repeat
the
it.
question. So you can repeat that, because then we'll make sure you understand.
What was the question? She was all right, say it again.
Hang on.
Give her the,
And
whatever she said
patient
was
with
right.
Sarah, because Sarah's going to get you the mic.
So we just want to clarify the reason the kidneys are so full of toxins and retaining metals is because the system that is supposed to be open, that goes from the hepatocytes to the gallbladder to the intestines, is blocked.
It's 2 steps. First, the system that's supposed to be draining from the liver to the gallbladder to the GI gets blocked. And there's a rerouting of excess load to the kidneys.
Through MRP3.
MRP3 dumps to the blood to go to the kidneys. The kidneys then have to suffer through all this load. And the kidneys get burned out if that load includes metals and lipopolysaccharides. There's probably other combinations that can do it. But lipopolysaccharides are very inflammatory, and metals have a modulating effect on the inflammatory response to lipopolysaccharides, in that when you have an inflammatory response, you have pro-inflammatory and anti-inflammatory cytokines all coming on at once to control it. But in the presence of mercury, you get only the pro-inflammatories, and you have an out-of-control sort of flame that happens. So first we block the liver, then we overload the kidney, and if we overload the kidney with the right things, we challenge the kidney to the point at which it stops working. And so when people talk about P-Tox, this is one of the things driving that. But I think there's a number of other factors that go into that. Is there usually a lot of ammonia? So there's a high catabolism. You've got a lot of protein breakdown, and you've probably got some damage to antidiuretic hormone generation. I don't know how that's controlled, but I'm sure we could find a
The other
pathway
thing
that
we
happens.
see in the kids with autism is high oxalate loads. And that is part of what's happening with the dumping. They end up dumping a bunch of oxalates and we can collect that in the urine and see that.
You get the needles and stuff. Yeah,
I don't know. If that
I
has
can't
to do
say
with-
exactly how that happens, but I've gone through being oxalate sensitive myself and fixing that. And the two things that seemed to fix it, one were binders that were more biotoxin specific. I think that there's a poisoning that leads to high oxalate production without moving the oxalate forward. And the other thing that fixed it was DIM. Can you give the mic to Dr. Cowden?
I have found that the oxalates are very often elevated in people who have major fungal overgrowth. And
I believe that.
Yeah, so I don't know exactly what's happening there, but if you get rid of the fungus, the oxalates do.
Yeah, so all of a sudden I have an oxalate problem. I have to stop. I'm married to a Mediterranean. It's like all I eat is oxalates all day. Yeah, and so I got to stop eating oxalates. But why didn't I used to have an oxalate problem? And so it was clearly something that was growing and blocking.
Yeah. Then one other quick comment. You said earlier that usually the phase 1 and phase 2 go together in those cells. There's one way that you can screw that up.
Well, phase 2 and phase 3 seem to go up and down together.
Yeah.
Phase 1 and 2 are supposed to, but they get easily dysregulated.
Yeah. Well, one way that you can really screw that up as a practitioner is to give things that drastically increase the cytochrome P450 activity without doing something that also increases the capability of conjugation.
Such as?
Yeah, well, years ago I had a patient that was a dentist who was putting in, using a lot of acrylates in his practice, and he started taking, without my knowledge, a lot of milk thistle. Milk thistle increases cytochrome P450 by 700% or 7-fold. And by doing so, he went into fluoride hemolysis and ended up in the hospital with 6 or 8 units of blood transfusion. Okay? Because he oxidized those acrylates, but he didn't have a conjugation to conjugate the oxidized
So he had
acrylates.
acrylic free radicals.
Yes, he had acrylic free radicals that were just chewing up itself.
Yeah, and that's I think that's a really go after milk thistle for the liver.
It's just another comment on the oxalate. So the oxalate issue, at least in my reality, is a huge issue in the autistic kids. And we found, last summit year I lectured on the chemtrails, on the spraying of aluminum in the sky, what goes up must come down. And we all, in the aphoresis sort of that we did over the last few years, we found that the average American has 100-fold to 200-fold higher levels of aluminum in the blood than any other metal from that. And there's plenty of plant research that shows that plants upregulate the oxalate production when they get exposed to anodized aluminum.
Oh, really?
And I have very strong proof that the same happens in the human body, because when we detox aluminum, the oxalate levels come down.
They come down.
So I think it's just an important thing to know from the fact.
Well, maybe it's a chelator for aluminum then. Aluminum oxalate, it should wrap around it real nice.
Yeah.
Like people use the malic acid oxalate's probably a better one.
Malate is weak silica.
Silica to push it out.
And that's,
So we'll do IV oxalate to detox from aluminum. Just kidding. Just kidding.
Silica,
That was a joke. I don't want anyone to pick that up as being real.
Chris, I'm sure you're still working on my liposomal silica.
Silica. I'm working on liposomal silica, I swear.
That's the solution.
Yeah.
Thank you, teacher Cleveland.
Yeah, the issue of dysbiosis and oxalates is a big issue.
Yeah.
Also, calcium dysregulation and oxalates, we see that as a big issue. So kids who don't, calcium doesn't go where it needs to go, so it binds to these oxalates. And then the issue of sulfur and oxalates, there's some kind of, I don't know if you guys can help us with the chemistry of the sulfur and the oxalate piece, but when the sulfur system isn't working, the oxalates are not detoxified.
It's hard to say what's happening first, because the people with calcium, a lot of them are high copper, low zinc. There's some other bigger dysfunction, and so the oxalate's probably getting pulled into that. And the sulfur system's all blown out when that happens too. And so what happened first? I don't know. But we'll make that the next project.
No, I just know we're talking about sulfur too, and I
Yeah.
want to.
Yeah, I mean the whole thing gets dysregulated. And when we get deeper into the sulfur system, you'll see that sulfur is really the core of all the things that we call signaling and the free radical signaling from ROS. It's mostly turning sulfurs on and off, and so there's not a robustness to the sulfurs. Then there's the whole system becomes out of order.
And I don't know if you know about like CBS upregulations, but I have a few slides on that. But that creates a big dysregulation in this whole pathway with glutathione,
Yeah.
and oxalates, and sulfur.
You got the methylation cycle and the
signaling and
sulfur.
everything like that.
Yeah. I mean, if you
That
that's
happens
being
in
able
Down
to.
syndrome kids, because I see a lot of Down syndrome kids, and trisomy 21, CBS is. with
There's
trisomy
CVS
21.
problems with that.
Yeah, so that's
Yeah.
where they upregulate CBS.
They upregulate CBS, and then they
The whole sulfur
create a
system.
sulfite excess and sulfite
And then
toxicity because SUOX can't keep up
Glutathione
with it.
redox is severely affected
Yeah.
in there, and so it's very similar to what's happening with the autism kids.
Yeah.
So that sulfur system, the redox signaling, the antioxidant, sulfation, methylation potential, all of that.
The whole thing's
Sitting where you're
dysregulated.
talking about.
Yeah.
You see that a lot in sulfite oxidase SNPs too, you know?
Repeat.
So I was just asking Hunter if she didn't see that with sulfite oxidase SNPs, where they, not able to convert sulfite into sulfate.
But we don't.
You don't always get sulfite oxidase on the
We
genomic
don't see the
panels.
SUOX SNPs that often, but the CBS is upregulated functionally in inflammation.
Yeah.
And it's upregulated functionally with like high testosterone, lead, aluminum. So even if you don't have a SNP in CBS, you have what they call like a functional CBS.
Yeah, I think the functional stuff is more important to measure. But then if that is upregulated, then you need SUOX to really flow and give a lot of molybdenum.
I don't have good scientific evidence of this yet, but a lot of the patients that have the pattern of sulfite oxidase problems, if you do a laser jet detoxification for sulfate drugs that are residual in the body, then all of a sudden that problem goes away.
Yeah, so this gets to this
Like from
this
back
epigenetic problem where you're not
So,
at the end we'll talk about epigenetics, and it's like the ways you affect epigenetics are really wild, and it seems a lot of them are energetic treatments.
the sulfur-containing drugs, Bactrim, Septra, even diuretics, furosemide, Bumax, etc., as well as the sulfur-containing diabetic drugs, all do this. And if you we can get these just from drinking water or bathing in tap water in big cities.
Yeah.
And so it's a gradually increasing problem.
Pause. Sorry.
Yep.
We have about 1,000 people online right now saying we can't hear the questions. So you guys all need to hold the mic right to your mouth when you speak.
You're
And
pretty
please
loud.
turn it off. Okay.
Can you turn it up?
That's Sarah. That's Sarah.
Maybe she can turn the feed up
Sarah.
because
Yeah.
it's pretty loud here.
So we'll turn the feed up. Whatever you guys want to do.
Well,
just washing dishes.
The feed to the digital.
So is that better now? Is that better?
You can turn that even more, I think.
What, is this how it's going online?
It's streaming right now.
Oh, through that mic? Is that how they get? Oh, okay, I got you. Okay.
Check it. The mic, it's important to hold it out like this. Instead of like this, it's like this. Okay?
Down the axis.
And then you need to hear yourself loudly.
Okay. Okay. Let's roll through this because we'll get to a little bit more of the punchline later, where we're trying to figure out where things have gotten blocked and what mechanisms unblock them. All right. Okay, so I'd like to, when I talk about detoxification, I'm talking about it within the scope of this big system of dominantly thiolic enzymes and enzymes and proteins. But we've gotten into this world where we talk about antioxidants, vitamin C and vitamin E and vitamin A, But we don't talk about how they actually go to work in the body. And the way they go to work in the body is by getting into the body's own network of antioxidants. And the body's own network of antioxidants is vastly more powerful than the ones that you can bring in. You'll never get to the redox potential of glutathione with vitamin C. But vitamin C gets drawn into this whole network. And in this network, you've got antioxidant enzymes like glutathione peroxidase that's going to use glutathione to quench free radicals, lipid peroxides, hydrogen peroxide. You've got thyroidoxin peroxidase, ascorbate peroxidase. That ascorbate peroxidase is using vitamin C as an antioxidant. You've got protein repair and regulatory proteins. You've got detoxification proteins. I think I've got slides on each one of these. And at the core of heavy, so we'll go back to that and we'll look at that because it's really core to what I talk about from a plant perspective and what Gary talks about from taking a pro-oxidant perspective, or what we talk about when, well, it's the same pro-oxidant perspective, when we do ozone. Same trigger, and there's a reason that they all do that. So for detoxification of metals, it's all surrounding glutathione, and the metals will bind to the sulfhydryl group on the glutathione, and that's the antioxidant part of the molecule as well. And in the glutathione system, it's important that we understand the enzymes are more important than the glutathione itself, if we had to choose. And so there's all these enzymes. There's things that synthesize glutathione. There's things that take it apart and reassemble it. There's the conjugation enzymes, the radical quenching enzymes, the ones that then re-reduce the glutathione after it's been oxidized for reuse. There's redoxins that use it for linking on and off of functional sites on proteins. And the redoxins and the concept of glutathiolation are where this whole story comes together with the free radicals and the plant-based chemicals for being able to do actual signaling in the body. And when we talk about antioxidants, again, I mean, I just told you this, there's things that you take from the outside like vitamin A, vitamin E, and vitamin C, and then there's endogenous stuff like glutathione, superoxide dismutase, CoQ10. Again, these are vastly more powerful than these. And then there's the machine that generates all the activity, and that's all these enzymes and proteins that we'll talk about. Oh, and then what did I not put in there at all, which are really exogenous, are the plant compounds. So polyphenolic antioxidants like green tea extract, pine bark extract, things that are in the exotic fruits like acai. These, we talk about them as antioxidants, but they're really not. They're stimulators of this super system, and they're stimulators because they create free radicals inside the cells. And then the sulfur compounds, we'll talk about the sulfur compounds like garlic, those really have strong free radical activity. So again, in these, you've got antioxidant enzymes that create all your antioxidant activity. You've got the reductases that bring it back into the central reduce pool. You've got thyroidoxin and glutaredoxin. These are putting sulfur groups on, and they're putting cysteines and glutathiones on and off of active sites, active sulfur sites on proteins in order to either protect them from some damaging scenario or turn them on and turn them off. So this is a major area that switching is taking place. So glutaredoxin, this on and off switch. So there's one way to look at it, that you have all these thiol groups. Thiol groups are very reduced. They're very sensitive to oxidation. And on the proteins, these tend to be very important active sites on the proteins. So if we're in a cell here, say we're in a cell, and there's all these reduced proteins here, and there's a little free radical generation. So we've got some mechanisms to quench that. We've got glutathione, we've got superoxide dismutase. If it's bigger than we can handle, we then start covering up the proteins, and sometimes we'll oxidate it and use them as an antioxidant. Other times we'll cover them up in order to protect them from irreparable damage. So we can take those sulfur sites, we can oxidize them a little bit, but if we oxidize them too far, they're irreparable. And so there's this system where we start covering these things up where there's a big free radical explosion going on. Thyroidoxin, I think we'll just confuse things. Just a little bit about the mitochondria. The mitochondria are these little animals inside of us, inside of ourselves. They say they were bacteria that we got to work for us. And they have their own unique chemistry. And even though they create all the free radical reactions to burn up your carbon and make energy, so they create the most amount of reactive oxygen species, the inside of them is the most reduced part of your cell. It's more reduced than the cytoplasm. And as such, it has special enzymes. Those glutaredoxin and thioredoxin and glutathione transferase, there's special ones inside the mitochondria. And they're designed so that they can handle little bursts of free radical activity, but the trade-off is that they're more susceptible to oxidation, irreparable oxidation, by mercury, cadmium, and arsenic. It's the way that their pKa is set up so that they deprotonate these more easily, so that they can go through these repair reactions. But the consequence of that is that they bind metals more easily. So that's why in metal toxicity we've got a lot of damage to the mitochondria. So we've got good working mitochondria and bad working mitochondria. Good working mitochondria are driving nutrients and oxygen into oxidative phosphorylation and all of our energy generation. They're creating ATP, and there's a little bit of free radical escape. Now, a beat-up mitochondria, and this is mostly damage around the membranes, is only creating a little bit of ATP and a lot of free radicals. So in fixing things, we want to repair the integrity of the system. And indeed, in autism, they see this is reduced ratios of reduced to oxidized glutathione. This was done by Jill James. And you have a more oxidized glutathione pool in the cytoplasm. It's running about 60% of the reduced ratio in an autistic kid versus a normotypic kid. But in the mitochondria, you see it even worse. You're only running at about 40% of the normal reduced glutathione pool. You've got a question coming?
You're getting good, Chris. These slides are better.
They're the same slides I threw you before.
Are they?
It was 8:30. The cocktails were out.
Sorry.
So you mentioned the term bad mitochondria. Could you clarify that?
Bad mitochondria. Is that clear?
How about naughty?
Is a damaged mitochondria. A good mitochondria is taking the fats that are coming at it, and it's creating ATP, and it's not creating a lot of free radical bleed. So the free radical bleed comes off the electron transport chain because it's called uncoupled. Well, this is a well-coupled electron transport chain. This is a poorly coupled electron transport chain where the electrons are draining out of the chain, not creating ATP, but they're creating reactive oxygen species.
Yeah, well, at least two mechanisms that I know of that will cause that bad mitochondria that you're talking about. One is if they can't produce enough coenzyme Q10. We know that the enzyme systems that make coenzyme Q10 in the body deteriorate over time with accumulation of toxins from the environment. That's why when you're 60 years old, you have a 60% chance of being CoQ10 deficient. When you're 80 years old, you have an 80% chance because it's a progressive accumulation over time. But the kids that have autism don't clear toxins as well as kids that don't have autism, so they're accumulating the toxins faster than the average person.
Or seen from another, I'll show a slide later, where the internal antioxidant system goes down with time.
Yeah.
And when that goes down, there's uncoupling of a lot of different things that are going on.
Yeah.
And so it could be toxins, but it could be, well, it's usually toxins creating an advanced degradation of the system. But in the autistic kid, I think there's a lot of different ways that we can decouple the system. In fact, I'll show you transgenerational effects where if the mother is poisoned, the child comes out with a diminished glutathione system out of the gate, and it'll stay like that.
Yeah. Well, one of the things we know is that all of the mitochondria for the child come from the mother. So, none of it comes from the dad. All the mitochondria comes from the mother.
Mitochondrial DNA all comes from the mother?
Mitochondrial DNA all comes from the mom, not from the dad. So the more damaged her mitochondria are, the more damaged the child's mitochondria are going to be. And so the less capable they're going to be of repair. So if you see a child with autism, you usually see a mom with autistic characteristics.
Yeah. Definitely.
Dana?
Hi.
Dana. Thank you, Lee.
Hey, Chris, I think I just saw a light bulb. Did you just learn something from legality? Did you?
Did I what?
Did you know that all the mitochondria came from the mother?
I did, but I'd forgotten that point.
Well, close enough.
I totally had forgotten. That was a light bulb.
Yeah, just a minute.
Just a bit.
Chris,
That was a firework, yes.
is it a? Can you comment, maybe not right now, on the NRF2 transcription factor?
Well, that's what we're leading up to.
Okay.
We wouldn't even be,
Thank
if
you.
there was no NRF2, I wouldn't even be standing here.
I understand.
Thanks, Robert. Just take it in the stratosphere.
That's
Just go
that's
right ahead.
right. Chris, would you get to the stratosphere already? Stop dawdling. All right, so the rest of this stuff, the phase 2 and phase 3 is for removal of free radical generating offenders, things that are creating problems. And incidentally, all the literature on that mostly comes from cancer journals because they see it as that system as being chemoprotective and getting rid of the things that are causing cancer. All right, so how does the system break down? Well, we've got genetic, where you've just born with, we've got genes that are weak. Then we've got epigenetic, where you're either born with your mother's dysfunction or you acquire a dysfunction along the way. And you know, I said environmental. Environmental can be epigenetic, where it turns things down. In fact, it's getting harder and harder to distinguish between what's an environmental insult that's just lowering the system, but it does that by changing your genetic expression of things. So it can be, and that's in glutathione, and glutathione has transferase both, genetic, epigenetic, or environmental. And then remember, phase 3 feeds all of phase 2 and phase 1, and so if phase 3 is blocked, the whole game gets blocked. This is what I was saying about age. You've got young and old rats, and you see glutathione and the antioxidant vitamins in the liver. Glutathione and the antioxidant vitamins going down 20 to 35% with age. Now, interestingly, in the enzyme world, the antioxidant enzymes lose their robustness, and the only thing that goes up are catalase and glutathione peroxidase. So the glutathione transferase, the glutathione reductase, the SOD, all these are tanking, so there's a lot more excess free radical damage going on, and so you upregulate catalase in order to accommodate this. Since we're talking about autism, let's just throw in one slide about age and glutamate and redox potential. So I said, you know, basically the antioxidant system's going down, which means the redox potential of the cell is going to go up. You're going to become more oxidized. And so these are neurons in a culture, and this is middle-aged and old neurons. This is the redox potential going down, so we want more reduced ones, and as we age, they get more oxidized. Now we flip this scale over, and now the top is more reduced, the bottom is more oxidized. This is embryonic, middle-aged, and old neurons. This is their normal oxidation schedule. But then if we throw some glutamate into the mix and hypercharge the glutamate receptors, we see an advanced oxidation schedule. So you decay much faster. So why is that? What do we generate when we create a lot of glutamate activity? We generate peroxynitrate free radicals out of superoxide and nitric oxide. And when we generate that free radical cascade off of the glutamate receptors, we create a lot of advanced inflammation in the brain. So this is the brain fog and the different neuroinflammatory things that are happening that are associated with glutamate excess.
What's the mechanism
The mechanism,
of glutamate causing excess peroxynitrate?
it happens at the glutamate receptors. Dr. Osman asked what the mechanism of glutamate causing peroxynitrate reactions is. And I don't know each step. It happens at the glutamate receptors, and when the glutamate is activated, they create peroxynitrate free radicals. And most of the stuff that I looked at already assumed that this was going on. I didn't go back into the mechanism of how that happens. But when you look at mercury toxicity, mercury neurotoxicity, its main reaction there, or its main neurotoxic mechanism, is through glutamate excess and stimulation of the glutamate receptors. In fact, in birds that eat fish, they found that the birds that were demethylating a lot of methyl mercury into inorganic mercury had the most neurotoxic lesions, and these were right at the lesions were right at the glutamate receptors. So there's something going on there.
Could it be like calmodulin?
I don't know.
Okay.
Wait, now we found something he doesn't know?
I don't know. Sorry.
Cam kinase is upregulated in kids like with pandas and pans where they have like Lyme disease or strep, and that causes a whole cellularly for the NMDA receptor to be turned on and glutamate to be turned on. So I don't know if
They definitely overlap. Because that's the glutamate receptor is the
free
NMDA.
radical excess microglial activation and free radical production.
That's the
there.
reaction that's happening. It's NMDA receptor activation, hyperactivation creates too many free radicals. All right, so where are we here? Oh, transgenerational dysfunction. Just so you know, I think we all know that epigenetics is passed generation to generation, but there was one study that I participated in, I did the analysis for, where they were looking at frogs. This is really relevant to autism, and they were trying to figure out what high levels of mercury do these frogs. So they took these two populations of frogs. They took the population, split them in half, exposed one half to mercury. So that's the F1 generation. One's got mercury, the other one didn't. They had their little frog babies, everything carried on, and they're like, well, that wasn't very exciting. So then they took the F2, the second generation, they exposed them both to mercury. And the generation where it was the second generation exposed to mercury, half of them died. So instead of generating resistance, it generated a susceptibility. So here we are in the third generation of having, 3/4 of the population with dental amalgam. That this may be a pretty serious issue.
The analogy is also with the Pottinger's cat study. Pottinger raised cats and gave the cats a junk food diet, and they became ill. Chronic degenerative disease. When he gave them a clean diet, then they recovered their health. If he gave the junk food diet to those cats and they had kittens, then the kittens would be ill. But if he gave them a clean diet, they would recover their health. If the third generation basically were given the junk food diet and then given a healthy diet after that, they did not recover.
They couldn't come back. So it was irreparable after a couple of generations.
Yeah.
Yeah, they needed some really serious therapy.
Yeah, so mercury is an important piece, but it's only part of the piece. You see, you got all these other things that are affecting your epigenetics.
Oh yeah. I mean, I'm not trying to be a mercurocentric man up here, but it's what I researched most. So my stories come from mercury. Yeah. And there's plenty more of them. I mean, this was here they exposed. These are fish eggs they exposed to mercury when the fish were born. There was an overall depletion in the antioxidant system. So the catalase, the glutathione peroxidase, glutathione transferase, glutathione reductase, these were all low, and it continued for the life of the fish. Now, the fish, in the absence of any pressures against it, could live through that. But when that's weak, now all the toxins come onto you, the soup of toxins come onto you, you can't handle anything anymore. So you make these little, very susceptible fish, and the same thing is happening to us. There is this transgenerational weakness in the system, so we want to figure out how to pop that thing back up. And I'd like to say I get to the end and I got the answer for you. All I got is some suggestions. And there's this was hamsters, same deal. The interutero exposure caused dose-dependent inhibition of development. Normally, after they're born, the antioxidant system blossoms in the brain. And they keep it kind of low, then they're born, and whoosh, it comes up, and it didn't come up in these ones that were exposed in utero. So when the child is developing and a mother with a lot of dental amalgams and a lot of other sources of toxins, you're going to have this situation where they're born, they may look normal, but they've got a depleted defense system. So they go in for those first 5 vaccines. Issue when they load up on them like that. Yeah, go ahead.
It's also true, I think, that the methyl mercury and the ethyl mercury cross the blood-brain barrier much more easily than the metallic mercury. So one of the things that's happening probably with these moms is they have the bugs in their gut that convert the metallic mercury.
It's a little bit different than you're thinking. They're making methyl mercury from the amalgam mercury and it's going up to the brain.
Yeah.
But what's coming off the amalgams, you've got 2 things. You've got the vapor coming off of them, which you inhale, goes into the lungs. That vapor goes right across the blood-brain barrier because it's not ionic yet.
Right.
So that's elemental mercury goes right into the brain.
Yeah.
Ionic mercury, Hg2+, does not go across the blood-brain barrier.
Right.
But once it's formed, it's the most cellularly damaging form.
Right.
More so than methyl and ethyl. So methyl and ethyl go across the blood-brain barrier and then at variable rates break down into inorganic mercury.
Right.
Elemental mercury comes off of the amalgam, goes right across and breaks down very quick into inorganic mercury.
Yeah.
I was mentioning a study with the fish-eating birds, and when they looked at the brain, they all had a ton of mercury in the brain, total mercury. Most of it was methyl mercury. But the ones that were demethylating at a rapid rate created the most amount of inorganic mercury in the brain, and that was what correlated with the lesions that were associated with the NMDA receptors. So when you're demethylating at a rapid rate, you're going to have more neurotoxicity for the same amount of methyl mercury. So when we look at our test results, you see someone with no amalgams, and if they have a lot of methyl mercury but not much inorganic mercury, they don't demethylate fast. We're not that worried about them. If they have a lot of methyl and a lot of inorganic, that's fast demethylation, they're going to have more neurotoxic methyl.
Would you dare to sort of translate that into that we should err on the side of hypermethylating mercury toxic people as a safeguard towards the destruction of the brain? You know, they're giving methylated folate and methylated B12.
Is this going to help? It's a let's dare here.
It's tempting, isn't
So
it?
it's tempting to say that because maybe you'll remethylate it.
In clinic,
I
it
would.
looks like that. In the clinic, it looks like that.
Yeah, and that's the big missing part of the map is where methylation protects glutathione processes. And in the way it's put forward by Amy Yasko and Ben Lynch, they have the CBS draining. They have basically methylation creating glutathione by creating cysteine from homocysteine. But once you get to the cysteine pool, your dietary inputs wash out your homocysteine recycling. And so there's some other feedback where methylation is supporting the glutathione processes and supporting detoxification of mercury. Is it through methylation of inorganic mercury? I don't know, but I would generally like that because methyl mercury is much more mobile. So if we put a bunch of methyl mercury into the blood, it goes into the brain according to its concentration in the blood. And then if we take it all out of the blood, it comes back out of the brain across concentration gradients. But once it's inorganic mercury in the brain, it takes forever to get out. We got to pry it off with a lot of activity of the glutathione system and pull it out. So if we could methylate it, that would be a good thing because it would flow back out of the brain. So it's possible those methylation enzymes do have the power to organic mercury, but in a test tube they can, like methyl B12 in a test tube methylates inorganic mercury. I used to make isotopically pure methyl mercury in grad school for experiments that we did using methyl B12. But it demands that the inorganic mercury not be bound to strong ligands like the sulfhydryls it's bound to in the body. When it's bound to those strong ligands, methyl B12 is not methylating it. So everybody was afraid to take sublingual methyl B12 because Hal Huggins said that it'll methylate the mercury in your mouth and go to your brain and kill you. We did an experiment on that. We had people with amalgams hold on to a bunch of aqueous, methyl B12, spit it out. Did we methylate any of the mercury? No. It's not happening because it's all kind of stuck to stuff. So I think it's a roundabout mechanism where it's keeping the robustness of the glutathione system alive, having proper methylation. I don't know that it's methylating the mercury in there. Yeah, I lost my cap. Go! So, what the glutathione system does is its robustness, and our glutathione levels go down. How does that affect the immune system? Here was an experiment where they tanked the glutathione levels in a mouse, and they looked at changes in TH1 and TH2, and intracellular immunity, interferon goes to the floor. Unfortunately, they did this by giving them a liquid ethanol diet. Not that I would ever experiment with that. But, you know, why do people, you know, get the flu or, you know, whatever after they have after they've got a bender weekend? Well, ethanol's tanking glutathione. When glutathione goes down, interferon goes down, but IL-4 goes up. IL-4 is humoral immunity. That's TH2. So this is going to make you more allergic, more food sensitive at the same time that you're more viral susceptible. That's a pretty significant outcome. Yes?
We actually did a study with Dr. Vojani on low glutathione and NK cells.
Oh, yeah.
And NK cells went down with glutathione and then came back up with glutathione.
When you bring the glutathione back up. So interferon does that too. And in fact, I don't know if it's in there. Somewhere in there it was, so this was when they tanked the glutathione, it went down, and when they brought it up, the interferon went right back up. Some of the other cytokines are a little bit slower to respond. And here again, this is then bringing the glutathione down using diethyl maleate, and this is the response of the different the different cytokines. So here's interferon tanking as the glutathione goes down. So you can see how a lot of these things are running together, and we get into this big spiral of immune dysregulation, creating lowered TH1, raised TH2, so you're becoming reactive to everything that comes around you. And in the case, again, of the NMDA receptors, Martin Powell writes a lot about how that glutamate excess creates multiple chemical sensitivities. So you've got chemical sensitivities, you've got food sensitivities, you're harboring a whole bunch of funguses and viruses, you've got the chronic infection, you've got chronic inflammation going on from that. Inflammation, we showed you, turns down detoxification, turns down expression of your internal antioxidant system. You start blowing out your vasculature, you're leaking lipopolysaccharide from your GI tract, you're leaking it through your vasculature into your brain, and all hell breaks loose. And here we are. So to cut to the chase, we want to turn back on the robustness of the system. We want to hit the triggers that like turn all the internal antioxidants on, turn all the detox on, and clear everything up. And this was from a talk that I gave, you saw at IAOMT, and this is getting away from the mercurocentric version of things. Like, a lot of these people come to you with that train wreck mix that, you know, we thought looked like mercury, but you test them, they don't have any mercury. And yeah, you know, if their methylation's all depleted, they do hold things intracellularly. But even if you get all that into circulation, it's not a massive amount of mercury. But they come to you and they say, I'm so mercury toxic. I was like, what's your source? You got amalgams? No. You eat fish? No. I got it from my mother. Well, it's gone, man. You know, you had like this much when you were this big, and now you're this big. You grew out of it. But then we go back and we see these transgenerational effects on the epigenetics. Your glutathione system doesn't work anymore. You've become susceptible to the soup of toxins. Everything hits you. And so you look like you're mercury toxic. You act like you're mercury toxic. You're certainly acting like you're mercury toxic. But you got no mercury. And so you guys know what I'm talking about there. So, all right, so how do we get in? How do we reset this system and get things to come back on? So phytogenomics, this is what I would always talk about before, is plants, using plants to upregulate all these good enzymes and proteins in the body. And we call this the anti-inflammatory cascade. And there's a couple classes that we use. Polyphenolic antioxidants, I talked about, pine bark extract, pomegranate extract. Then the sulfur compounds also do this. But what else does this? Pro-oxidants. So how do they do that? This is the NRF2 protein. We're there. We've caught up to Robert.
Yay!
We're out in the cell. And we've got this complex here, the KEAP1 NRF2 complex. There are two proteins, and the KEAP1 appropriately keeps the NRF2 in place until something tags it. What tags it? It says enzyme inducers. So enzyme inducers tag this, the NRF2 travels into the nucleus, and all the genes with a certain promoter region called the antioxidant response element unfold, get transcribed, RNA comes in, they make the RNA, and you make those proteins, you make more glutathionase transferase, you make more superoxide dismutase, more glutathione, and you've got molecular chaperones, antioxidants, phase 2 enzymes, and you lead to cell survival. All right, now what are the enzyme inducers, and what's going on here? What do we see? Sulfhydryl groups. Sulfhydryl groups, what do? They go from being sulfhydryls to being oxidized disulfides. I'm sure this is SS. They only have one S, but there should be two. They oxidize them.
Where is that?
This is in,
It's in the cytoplasm.
here's the cytoplasm, that's the nucleus.
Yeah, but what's that?
Actin, it's an actin polymer.
Just floating in the cytoplasm.
Floating in the cytoplasm. It's not membrane associated. And so in my normal talk about this, I talk, oh yeah, here's our polyphenols, because I use polyphenols more than sulfur compounds, because the sulfur compounds tend to irritate people more, and I'll tell you why in a second when it all starts to come together. So green tea extract has epicatechin. We've got ellagic acid from pomegranates. Here's the one I use the most. Often I'll flash a picture of the medicine Buddha and say, I use medicine Buddha. Well, the medicine Buddha, there's his hand. He's holding a plant. This is sacred to Shiva in Ayurvedic medicine. It's called the king of herbs in Tibet. It's called the Myrobalan or Haritaki. And in Haritaki are all of the great, do I have them on there? All of the great polyphenolic antioxidants. There's epigallocatechin, catechin, gallic acid, luteolin, all the stuff we want. Nice blend of things. And I showed you earlier this story of the young and old rats. And the old rats, the robustness of the antioxidant system has gone down. And so we feed them this aqueous extract of the Haritaki, and you see the antioxidant enzymes in the salt all come back up to what they were when it was young. So the old rat gets young rat antioxidant enzymes. And remember I said the catalase and glutathione peroxidase were upregulated to accommodate that. They go back down to where they were. So there's a normalization of the system inside.
It's important, I think, for the listeners to know that this is also researched as a biofilm record. So it's important to know that it has a dual effect that makes it almost an ideal kind of herb for
so it's used in Chinese medicine. It's used in Tibet for everything. all their formulas have it. And in Chinese medicine, they use it to rebuild the epithelia of the GI tract. And a lot of this is because of this biofilm breaking activity that the Haritaki has. And in Ayurveda, it's part of Triphala, their main digestive tonic, which is Haritaki, Bibitaki, and Amalaki. Triphala. All right, so here we see an enzyme inducer that turns on the NRF2, fixing the old rat, turning him back into a young rat, at least from an antioxidant perspective. Andrew, did you have a question? Did you want me to go back?
What?
The NRF2 slide.
Well, we're coming back to it.
Take your time.
So you just go through the enzyme. Can you go through that one more time?
Okay. We'll keep these off to the side. All right, in the cell, you've got in the cytoplasm these two proteins. And they're holding on to each other. And certain, quote, enzyme inducers come and tag this keep one protein, which changes conformation. It goes from here to here, and that releases the NRF2, like the Nerf ball here, and it travels into the nucleus, across the nuclear membrane into the nucleus, and it triggers a transcription of a family of genes that all have the same promoter region. Promoter region is like a big switch, and it's called the antioxidant response element. Hence, it turns on antioxidants.
Wait, I got a question from Terry. I don't know if we can see a little bit of a. Can we stand up for a second? Get closer. That's really talking.
This might be addressed to.
Oh, there we go. Go ahead.
This might be addressed to Dr. Usman because Dr. Klinghart mentioned breaking biofilm, and before willy-nilly breaking biofilm, are there some words of caution that you would want to give about breaking up biofilm?
I think there's probably a lot. And now we segue into Andrew's lecture.
Yeah.
Yeah.
I mean, that's a good point, Terry, that we don't want to go willy-nilly breaking down biofilm because what's underneath the biofilm are toxins, metals, bugs, and as they're released, we're going to get an inflammatory cascade being produced. And if we don't have these systems in place first, we're going to cause a big problem.
Yeah. That was very succinct. So fix the native antioxidant system, then start letting all hell break loose. Or then start opening up the nest.
Yes.
Okay.
It is. It's a nest.
Yeah. So what are these enzyme inducers? We've got the phytochemicals that can do that, and I'll show sulfur chemicals in a second. I showed some polyphenols that do it. And I said it's really because once they got into it, the researchers were calling them antioxidants. And then once they really got into what was happening here, they saw that epicatechin that we thought was a great antioxidant from green tea is coming down here and creating a little free radical cascade that's oxidizing those sulfhydryls together, and they're releasing the NRF2. What else can do it? Reactive oxygen species, and I'll show you some pictures of papers, ozone therapy. They find NRF2 goes up when they give them the ozone. NRF2 goes up when they give them the hydrogen peroxide. Reactive nitrogen species. So they find that peroxynitrate reacts with glutathione or with cysteine, and it actually creates an upregulation of NRF2. It goes into the cell, and it's super efficient at creating this oxidation. So here, an antioxidant, cysteine, combined with an oxidant, peroxynitrate, is making an enzyme inducer to turn on the NRF2 in order to clear out the oxidant threat. It is hormesis. Hormesis is, I go poke, poke, poke until I get slapped back. Well, that's one way to say it. It's irritating the system until the system bounces back and creates something to clear out the threat. In fact, there's an anti-aging researcher at the University of Colorado that I talked to, and he goes, the only thing, we've tested thousands of molecules here for extending lifespan, and hormesis is the only thing that actually does it. And irritating these animals is the only way to make them live longer. So hard. Life is suffering, so you stay alive. That's why, rich people do. No, all right. We don't even
Chris,
go
do you have
there.
another slide on that switch? The hormetic switch, do you have another slide?
I don't have the slide that you want.
Okay.
You're going to make the slide that you want. So reactive oxygen species, reactive nitrogen species, reactive sulfur species. So you create an oxidant, and an oxidant is passing along oxidation and oxidizing other things which are turning into radicals. So even if you bring in ozone, you may create oxidized nitrogen, and oxidized nitrogen will come in. You may create oxidized sulfur, and oxidized sulfur comes in and hits the switch. And now, I found papers where they're going through catalogs of molecules and seeing what turns on NRF2. Guess what? DMPS and DMSA do. maybe all these kids that are coming in, we're like, just get a little more mercury out of them, they'll get better, and you keep jacking them up with DMPS and DMSA, you're hitting NRF2. Maybe some of the kids that we fixed with chelation therapy weren't metal toxic. They needed a reboot to the whole system. Kind of answers a lot. But, yeah, well,
Especially when we don't see metals coming.
yeah,
And they get
but they get better. You know, well, the metal was on this super sensitive site. Oh, maybe not at all. Maybe it never had anything to do with metals. Just in case you thought I was mercurocentric. Maybe it has nothing to do with me.
Got it.
Oh no! I thought it was me. All right. Sulfur compounds. So garlic. And you know, back in the day, how many times did you hear, oh, you use garlic for metal detox because it's a sulfur compound and therefore it's a chelator, right? Didn't we say, or at least some people would say that. It said it's a sulfur compound, it's chelating you. And all the sulfurs anybody took were always chelators. They're chelating you. These are free radicals, man. These things are really, that's why they kill bugs. That's why garlic kills things, free radical reactions. And the free radical and the sulfur chemicals are stronger than the polyphenols for inducing NRF2, a fair amount stronger. But we went into using them for people, and we found that it would create more cellular stress. Well, probably because they're free radicals, because they create a lot of free radicals, but secondarily because of these CBS problems where they can't process. Most of the toxic people can't process sulfur right, and so CBS gets upregulated, and they're trying to shuttle everything out ultimately as sulfate, but it gets stuck as sulfite, so you got sulfite toxicity, and then all the problems around sulfite toxicity, so you give them what?
Yeah. And biotin.
And biotin. Molybdenum and biotin.
And B1.
You give them the core, because they've got all of it.
Give them core, yeah. Oh, so core wasn't, and core was originally KPU, HPU stuff, but it turns out it's also SUOX stuff, or maybe it always was SUOX stuff, and HPU, but it was just the sulfite toxicity. So it's like, this is why I want to get the mechanisms right, because we go, you know, we create something, we know it works, we give it the wrong attributes, and we can't take the next step. Just use it works. You know, if we know why, then we can evolve a little bit better.
So what about B6? Wouldn't that upregulate that? With the KT? Can you repeat that question?
Would B6 upregulate CBS?
Nice.
Yeah. Does it do it real strongly? I mean, it turns methylation cycles.
I mean, CBS and cysteine beta lactase, whatever that is.
Cysteine beta synthase and beta lyase.
The beta lyase, cysteine beta synthase, cysteine beta lyase are
B6 dependent. So,
but they
well,
need
there,
B6 for the
yeah, I would, you know, if your B6 is causing you problems, then it's probably, unless it's peripheral neuropathy, if it's causing problems, then you probably need these other things. I would, you know, not shy away from the B6, just take the other stuff you need. Okay. Well, so like I said, the sulfur stuff is stronger than the polyphenols, but people run into problems with it because they're stronger free radical generators, and people need you know, their sulfation system supported. So we know most of these things in this reactive oxygen cascade can trigger NRF2 to go in, turn up all the goods at once. This would be a little bit hard to follow. It's probably a little deeper than we need to go, but there's this whole system involving glutaredoxin, putting glutathione onto active sulfur sites, or putting cysteine onto active sulfur sites. They used to think it was just to the sulfur sites from excess oxidation. Like I said, if you take it too far, it's irreparable. If you take it just a little bit, you can repair it. And so there was this system of putting them on and off, but it was actually kind of hard to get raw glutathione onto these things. But what they found is that when you oxidize the glutathione with either oxygen or with nitrogen, and you create these are proteins with oxidized proteins, and that's oxidized glutathione. Those are both oxidized glutathione, but this is very strongly oxidized. So they found that when the free radical stress oxidizes the thiols a little bit, you get the signaling reactions even stronger. And one of the signaling reactions is to go in and tag that to turn on the NRF2. So these pro-oxidant therapies do have a nice direct road into upregulating things if the system actually can do it, if the switches actually work. And these are all proteins that are regulating redox reactions by getting oxidized or reduced to turn them on and off. And these are all major proteins. And notice most of them, it's thiol groups that are getting oxidized and reduced. It really is that thiol system that is doing the on and off. That is the basis for all of this. And then you're going to irritate it with different things that you bring into the system. So this is an article on ozone therapy, and they call it a mild oxidative stress. And they talk about these two different ways that oxidative stress can go. They have mild oxidative stress, turning on the NRF2, bringing up all of these chemoprotective things that are in the end anti-neoplastic and lead to survival. And then they have severe oxidative stress inducing nuclear factor kappa beta and creating runaway oxidation. They go on to say it in sort of a similar way.
On the slide before that one, I think it, no, right before that. Yeah, there. Okay, yeah, so you're saying down at the bottom that the heme is not modified. So it's not just all, left column.
Yeah, heme being modified by nitric oxide. You've got a mitochondrial complex, and you've got guanylate cyclase, which I don't know what that does. And so different things are either activating or inhibiting different molecules. That's by targeting heme.
Yeah.
And all these others are thiols on these proteins that are targeted sometimes by reactive oxygen species, sometimes reactive nitrogen species. There's hydrogen peroxide.
Yeah.
Here they have electrophilic lipids, which means oxidized lipids. Like if you ozonate a lipid, it'll be an electrophilic lipid. Here's species. There's reactive sulfur nitrosothiols, so putting nitrogens on thiols. More reactive oxygen species. There's superoxide. So these are all different free radicals, but they're not all oxygen. So everybody, in the chlorine dioxide game, they all talk about reactive oxygen species. But all this signaling that's going on is cascading through nitrogen, oxygen, and sulfur groups. And again, they're talking about mild oxidation versus severe oxidation, but I think it's the right kind of oxidation. I think that's what we're going to move towards understanding more. Hopefully Gary will shed some light on right versus wrong oxidants. That's where I think he's going to be going with a lot of this. But again, it's targeting this response from the cell. Oh my God, clean everything up. And of course, we see that having an NRF2, there's so many papers in it, and where they knock things go wrong. This is NRF2 inhibiting methylmercury, creating essentially Parkinson's disease. This is a Parkinson's mouse model with dopamine neuron degeneration. And if they knock out NRF2 and give it methylmercury, it's Parkinson's immediately. If they keep NRF2 in there and upregulate it, they can handle a lot of methylmercury without inducing the Parkinson's. The brain, remember I said you've got to have this stuff everywhere to get rid of it. These are to get rid of toxins. These are capillaries at the blood-brain barrier. Stained in red is phase 2. Stained in green is phase 3. Stained in yellow, they're co-located. So the whole blood-brain barrier is studded with this whole machinery to act as an export pump or a free radical consumption machine. So if that's right, why is it so hard to fix so many people? So what if the mechanism isn't working? you switch something off, but you can't switch it back on. And there's a lot of examples about of this. Let me go into them more. And you're going to see a lot more papers coming out with a lot more ways that you can block NRF2 transcription. The most obvious one right now is mold toxins. This is ochratoxin. Is ochratoxin an NRF2 inhibitor? There's really good data for it. Reduction in antioxidant defenses contribute to ochratoxin A toxicity, and if you read down through it, it's through blocking NRF2. NRF2 increases survival and attenuates alveolar growth inhibition in neonatal mice exposed to hyperoxia. So, we're in pro-oxidant therapy. We need to be able to respond to it right. It's a hormetic thing. If they knocked out NRF2 and they exposed mice to hyperoxygen, they don't do well. It's working, no problemo. So, we got people say they got ochratoxin, mold toxicity, NRF2 inhibition, and we send them in for ozone therapy. They might not do very well with that. Peroxide therapy, they may not do well with that. Chlorine dioxide therapy, they may not do very well with that. And there's a heck of a lot of cases of people not doing well with that. And so there's a hormetic switch, and the hormetic switch is broken. You've got to nurse them back up and get that switch back on. It's a question of how you do that. I don't know everything about doing that, but there was one beautiful paper in, remember I was talking about prostate tumors, and the glutathione transferase is turned off epigenetically in prostate tumors? Well, that's because NRF2 is epigenetically turned off, and they restored the activity of it with diindolylmethane. Yeah. So diindolylmethane. Diindolylmethane is known to be cancer protective. It's mostly thought of as for women, but it's one of the components of broccoli. It's not a sulfur component. It's a nitrogen component. There's no sulfur in it. And so people who have sulfur issues will do better with the DIM. But it looks like it was taking away the hypermethylation of the promoter region and restoring its natural function. So we're going to find more tools like this to unwrap the natural function of these people that we give these hormetic things to, pro-oxidants, or we give them lots of sulfur and they have really bad reactions to it. We give them molybdenum, they still have bad reactions to it. How are we going to peel away this epigenetic problem? So DIM is one of these things. And, you know, this was an epigenetic problem. I don't have a good before, but this is back when I was using polyphenols. And my son, this is an epigenetic thing we had in my family. My mother got warts all over her hand when she was pregnant with me. Then I got them really bad. Then my son got them really bad. It's a simple little virus. You know, everybody who doesn't have them is killing it, and everybody who does isn't. So they don't know what it is, but that's an obvious epigenetic dysfunction. And he was, you know, you don't see many here because they'd already come off. Like, this is 2 years of trying to fix him, giving everything at reasonable rates. You know, he's a 6-year-old. I'm not going to give him a high dose. You know, I'm his father. But I gave him everything I had, and I gave him every naturopathic trick in the book. I gave him everything, and then I started the torture, you know, where you start burning them off. And the torture didn't work either. And then finally, I started understanding these threshold doses you need to really invoke things. And so I gave him 3 times the adult dose while he was 6. And he was up for it because he was, you know, kids wouldn't shake his hand and stuff. So I gave him this. I left for like 6 days. I'd come back and I say, let me see your hand. And this is what he showed me. There used to be like 30, 40 warts along there. They'd come up here. They were all across here. You know, anywhere you see red used to be covered in warts. And then that's a week later, and now we're 4 years later. Nothing's ever come back. But it was a threshold dose I had to get to wipe that clean. So how do we get to the threshold doses without, you know, causing too much damage? Last thing I want to show, just so we can start thinking about how to use our tools a little bit more accurately. This was St. John's wort, and this was for upregulating the same kind of enzymes I like to talk about. This is phase 3, and that's phase 2. So that's the transporters, that's glutathione, that's transferase. Like I said, there's got to be a threshold dose. So this was no St. John's wort, low dose, medium dose, high dose, really high dose. And so here you get nothing until you get to the high doses. Here it's a little bit more gradual. So you need a threshold dose, and then this is probably the most important of all of them, is that there's a time-limited hormetic response. So they took that highest dose of St. John, these mice that dose for 30 days. So in the first 10 days, that's 100% of this expression of transport proteins, that's glutathione, that's transferase. 10 days, we go up to a max, and then what happens over the next 20 days? We go back down to baseline. The body stops responding to the input. And both phase 1 and phase 2 did that. Phase 2 did it faster. So that says a lot about how we time doses, that we should pulse things. I always related it to my experiences in Chinese medicine, where I was in grad school all beat up. I got deficient kidney, excess liver, and I'd go into this doctor and he'd give me a pile of herbs, old school, big pile of herbs, cook it down into 1 cup, and you do it for 5-6 days, and after like 4 or 5 days, it's just like you're reset. They don't say, oh, take this every day for the next 20 months. Certain nutrients you need to support the system, those are daily irritants. I don't think are supposed to be daily things. So this could be another reason why some people crash and burn with like CD therapy. If they go, one, either their NRF2 switch isn't working, they've got that epigenetically hypermethylated, so you can't induce those genes, and then you're just pouring in pro-oxidant and giving them oxidative stress. 2, you keep a constant dose for too long and the hormetic mechanism stops working, and you go from provoking a response to just burning out the body. But either way, in the end, it's the same thing that you end up with.
Just quickly, do you think that the 10-day period may be true for many other things also?
Yes! 10 days is that's my go-to. I start them at 5 days 2 off, because you get up halfway up to max. It might be as much as they can handle, because you're starting to mobilize a lot of stuff. Then I give them some time off. After they get deeper in, this is for the mercury detox protocols. Once they get deeper into it, I flip them over to 10 days on, 4 days off. But I think 10 days, I think that's a real nice target. And if you give them 4 off, it works in 2-week pieces.
Yeah. When I was doing developing the counter support program for Lyme disease, I noticed that if I went longer than 12 days, they didn't do as well. If I stopped like at 12 days, then I had to give them a couple days off. So.
Bingo. So I think that's something that, if we're here to create consensus, I think that's one of the things. I think, what pro-oxidant therapies are doing for us and how they overlap with a lot of plant-based therapies, hitting the same triggers. I think that's a real important thing, and that those triggers don't always work, and we got to figure out when they don't and how to fix that. And if you go a little farther out, wildness, I fixed my son's warts by a high dose, finally flushing everything off, whereas low doses weren't doing anything. And that could be a key also in the pro-oxidants, that you've got internal low-dose pro-oxidants going every day, causing you problems, and then you take, a shot of, say that switches do work, then you take a shot of ozone or a shot of ASEA or peroxide, and boom, it's enough to wake the system up and turn on the hormesis switches, whereas, your bleeding, daily bleeding mitochondria aren't doing it, they're just causing stress. And so if we could, accurately get in and figure out what the thresholds we need to do to get it working are, what the signs are when the switches aren't working, and what we need to do to fix those. Because you look at warts, I mean, witches charm those off. Homeopathics work on them. I mean, what are the different ways we can affect this epigenetic change?
I just wanted to mention that Dr. William Hulse, he's a famous osteopath. He also taught Gary and myself. That was his protocol, was 10 days on, 4 days off for parasite detoxification. Interesting.
That's what we use for Artemis.
So in his career, that's what he learned.
10-4.
The way to do it. Interesting.
And that was where I did it, and he was talking about Dr. Gary Gordon 2 hours.
So that's the extent of what I wanted to put out. Oh, wrong computer again. That's the extent of what I wanted to put out there onto the floor to talk about. Thank you, Dana.
Thank you. In this room, and then can you see if any people have questions that they can just write away? So if you're in bottoms up land and you can get it through the next 12 seconds, we might answer your question live. Tell them something good.
I just heard the presentation. Amazing.
Okay, so now go back to the hormetic switch, because as a mom, that one talks to me.
It's probably the biggest one we got to figure out. I mean, first it's seeing that it's there, and that we're approaching it from different ways, but then figuring out what breaks it and how we fix that.
But what is it? Like, try to define it.
What is it? I mean, you're triggering your body to say, oh my God, we got to shift gears and turn into house cleaning mode. These used to be called house cleaning proteins. You know, the selenoproteins like glutathione reductase, or glutathione peroxidase and thyroidoxine reductase were called house cleaning proteins before they gave them real names. So you're going about your business, you're just working all the time, doing your thing, and then somebody smacks you in the head and you look around and you realize that your house is a shithole and you got to clean it up. And it's kind of like that. So something triggers you to clean up and get all the bad actors out. Maybe you were so tired that you weren't realizing how much they were building up. Again, if you look at that St. John's wort dosing, you got used to it. You ignored it after a while. And so how many things do we ignore? And I think they're building up at slow rates. And so we need things that pique the interest of our cellular biochemistry and turn on these house cleaning genes and clean things up. And again, it's not just detox, because if you bring glutathione up, what comes with that? Innate immunity comes back up. And so it's house cleaning of the intracellular bugs as well.
Yeah, well, when you do this turn-on switch, you're getting that oxidative burst also, which will get rid of not just bugs but also toxins. So you're turning on that phase 1 system by that oxidative burst.
Yeah, and you think that's gonna, because somehow, it's like when they talk about ozone therapy, when Frank Schallenberger lectures on ozone therapy, he says, oh, oxygen utilization potential is down, we give them ozone and it bursts back up. Well, you're not using the ozone. What are you doing? I mean, what is it doing? And that's why I like that picture of the weak mitochondria versus the healthy mitochondria. And maybe it just tips, it either makes the right species to hit the switch to reset and clean up, or it goes over a certain threshold at which you reset and clean up. And I think when you reset and clean up, you recouple the electron transport chain, and that gives you a burst, that gives you the higher energy.
Yeah. If you got a bunch of sick mitochondria in the cell, you're just going to have a bunch of sick mitochondria in the cell. But if you do something that causes the really sick mitochondria to go ahead and die, then you're going to be replacing those mitochondria.
Yeah, and somewhere in there is the exact mechanism. I don't think we have it fingered yet. Gary, do you have more? No. We got to figure out what those triggers are so we can more accurately use them, because obviously it's stimulating the mitochondria, but we don't know exactly how.
Chris, would you go over the products that Quicksilver has and how they address the different sources?
Sure.
Don't miss that part.
Hey, Robert, there's a bunch over there next to you. Can you bring me? Do I have slides? I do. I keep going to
Can
your
you touch
computer.
that computer one more time?
All of it.
Okay,
Sorry, I didn't touch it.
I was looking at the pretty one. Okay, while you're finding slides, because I know you're a man that can multitask, you're just that kind of guy. I can be heard, I know I can. Okay.
Here you go.
Oh, God, they're probably going, no, don't hit Dana the ball. It's too loud. Okay. I'm going to describe a parrot.
Thanks.
They're doing a protocol to kill parasites. Are you with me, Chris?
You got a C like poet over there. Yes. Protocol, killing parasites.
For kids with autism.
Yes.
They've been doing it a long time. It involves chlorine dioxide and a bunch of herbs to kill parasites at the full moon.
Okay.
They do. The kids really like the brain clarity that they get. It really looks like they're going to hit it out of the park. And then it starts going the opposite direction, but not enough that they cold turkey know to just do something different.
Right.
They keep pushing, keep pushing. Their heart is set on this really being the solution for them. They continue with the P-Tox that we talked about, with it peeing all the time, and they push harder. They do more CD. They do more of everything. And we have some people who've gotten to the point where they quit, and they're way worse than they were before.
Right.
I don't need to talk about that. When they're hitting those thresholds, if they start to shift from it being a positive to a negative, what do you think is happening, and how long would you wait before you would shift?
Well, that's a pretty broad question.
The moms get it. What's your problem?
Well, it demands a redirection. Why don't I go through the products and then we'll go, because I want that to be a more.
I like it.
It's
Perfect.
a tease. Yeah.
That works.
We'll talk about this, and then we're going to talk about that because it's obviously the crucial question that goes on here.
Well, it's a big deal.
Yes. It's pretty much why we're here. So our product system, I'd said, if I go back one slide, I'd say there's three main things that you need for moving metals out of the system. And in the basic glutathione upregulation, that's got to be mercury, cadmium, and arsenic as your main ones. You need phase 3 clearance, that's the transport proteins bringing things out. You need phase 2 activity, that's pulling metals off of the cellular proteins onto the glutathione, and you of course need the glutathione.
Take your time.
So in our system, this is what we use for phase 3. For phase 3, we clear metals away from the lining of the GI tract. So this product here, IMD, are little silica particles that are saturated with sulfhydryl groups. So we graft onto the silica particles. These are not absorbed, non-absorbed silica with little carbon chains grafted onto them that terminate in sulfhydryl groups. So you're taking loads of sulfhydryl groups on a little particle through the GI tract, and it bounces around there and grabs all the metals and pulls them away. One of the things when you go into the papers on the chemistry of the transport proteins is when they looked at them transporting metals, yeah, they transport metals, but they slow down when the metals are around. So if you're not always clearing the metals away from the proximal end of the transporter, you slow down the process. So as you move them away, everything starts to flow back down to the GI tract. In fact, this was the first product that we made, and I made it to pull myself out of my own mercury and DMSA-induced haze, and by clearing all these metals out of the GI tract, I felt better. And I went and lectured on it on my 40th birthday at THRiiiVE's Chemical and Heavy Metal Detox Extravaganza, and he was immediately like, give me that. And like I said, filled a hole in all of our arsenals. And it was doing what chlorella does, but one scoop of this was like 100 chlorella. And, this does lead into the whole conversation about what different binders are. And binders are often spoken in too broad of a term, as if they all do the same thing. But we've got a world of different chemicals with a world of different chemical personalities, and each binder associates and attracts and associates with different personalities. And so you've got clays, and you've got zeolite, which is a super clay. They have a certain type of absorption capability, but they don't bind metals. They don't have any thiol groups. You've got carbon, which has got the broadest range of affinity, and even will pull metals in just because it pulls everything in, but it doesn't have actual covalent binding with metals. You've got alginates and pectinates, and those have more of an immunomodulatory effect on the GI tract. And as we've seen, when you're inflamed, you shut down transport. But if you can calm down that immune reaction, you'll transport again. They don't have much strong binding affinity. Maybe a little bit for lead, but they're not doing anything for mercury, cadmium, and arsenic. Then you've got chlorella, which has got a lot of different binding types. It's got anionic, it's got cationic, it's got thiol. And just to retrace a little bit, anionic would be cholestyramine. So cholestyramine is binding these products of phase 2 conjugation. So when we put glutathione or glucuronic acid or sulfate onto a molecule, we create an anionic charge. And that's water soluble, and it goes through these transport proteins and gets down to the GI tract. And if you have an anion exchange resin in there, it will attract and absorb those so you don't reabsorb them and enter hepatic circulation. That's what cholestyramine does. And I'd mentioned earlier that the natural version of that is chitosan. Chitosan are repeating chitosan, C-H-I-T-O-S-A-N. And both chitosan and cholestyramine are incorrectly called fat binders. And what they actually bind are the biosalts that are emulsifying the fat so you can absorb it. So chitosan is used for weight loss because it blocks you from absorption of fats because it picks up your bile acids. What I failed to say earlier in phase 3 and draining from the hepatocytes into the gallbladder, that it's the same transporters that move the bile acids. So if you have cholestasis, you have toxostasis. My latest thing for draining the gallbladder, I made a great methylation sublingual, and you'd see the liver activities, and it would sink the gallbladder activity. And some of the first people I gave it to got nauseous, and I said, "You just ate something bad here at the conference." So I saw you turn up the liver, the gallbladder has to be tonified and turned up with that. That's the transport system for the toxins. This is part of why Hal Huggins would always have you eat butter all day. The butyrate helps heal your gut, but the fat all the time gets the gallbladder flowing. So if you have cholestasis, don't try to move toxins until you move the bile. And then
Is this why people get constipated when they use cholestyramine?
yeah, that's probably a big part of it.
One of the biggest stimuluses for peristalsis is the bile salts in contact with the mucosa of the intestine.
So yes, absorbing the bile salts.
I have been very aware for many years that in Europe they use beta-sitosterol as an exact equivalent of cholestyramine. And there's a lot of it in stabilized rice bran, which is what I've been using for 10 years. And so just realize beta-sitosterol has been documented to give the same effect as you get from cholestyramine, but sure you got to stabilized rice bran is kind of cheap. It's been around for years. So anyhow, that's what I live on.
Okay, good to know. So there's another binder, in the stabilized rice bran for biotoxins, and so the cholestyramine, the chitosan, the rice bran, all good for the biotoxins.
No, they're binding the bile salts. The bile salts are actually reabsorption of certain fat-soluble toxins from the gut. Like the mycotoxins, many of those are fatty molecules. If you look at them, the chemistry.
They're big. I was associating it with them being conjugated in phase 2, pushed out through the liver, and then getting deconjugated. So the glucuronic acid, like for methylmercury, it comes in as methylmercury cysteine that you digest from the fish, and you think that it's methionine. So you take it in on methionine transporters, conjugate it to glutathione, push it out, you break down the glutathione in the GI tract, so you're left with methylmercury cysteine, which you reabsorb. So the mycotoxins coming out conjugated, then deconjugating and being reabsorbed. But it could be a secondary effect that the bile salts are helping with the reabsorption. Because if they're still conjugated, they shouldn't reabsorb. But if they deconjugate, then they'll reabsorb.
Right.
But if you bind them onto something
right.
and stabilize it on a solid phase, then it probably won't deconjugate.
That beta-cytosterol story really started in Europe, where they approved it years ago for the treatment of type 4 hyperlipidemia, specifically for its documented ability to drop LDL and really lower triglycerides. So it really does have a measurable, significant effect on lipidemias.
Yep. And so the last part of the binder story is, so you go to chlorella. Chlorella's got some anion exchange, some cation exchange. It doesn't have a lot of thiols. And then if you're really targeting metals, you want something that's just thiols. And so these are silica particles with just thiols. There's enough thiol on here to hold 200 milligrams of mercury on a gram of this. That's a lot of mercury. 200 milligrams of mercury is enough to kill you. So you got a lot of binding capacity, and as you grab all that and pull it away from those transport proteins, the transport proteins then free up and start moving again. So this is the first thing that we started doing, and that has a tremendous effect on its own, but then we want to go and we want to upregulate phase 2. So how do we upregulate phase 2? We looked at a lot of different ways to do it with the polyphenols, with the sulfur chemicals. The sulfur chemicals that we use is lipoic acid, and we use R-lipoic acid. I think I have a couple slides on that a little bit later. Specifically because in that NRF2 coupling, S-lipoic acid does not turn the switch, but R does. So alpha-lipoic acid is equal amounts of R and S, just like D and L amino acids. Only L works, D doesn't. So alpha is R and S, but only R turns the switch, but they both fight to get at the same spot. So S is kind of holding you back. You go with R, you're far, far more potent. And then we use the polyphenols. And then we do a lipid-based liposomal delivery. We're doing sort of hybrid liposomes. Liposomes are coming forward at a pretty rapid rate. And there's some things that you need to have a really good liposome that'll absorb real well in the oral cavity. And what you really need is the right size and secondarily the right membrane terrain. But the right size will get intraoral absorption, as well as absorption as you swallow going down the GI tract. You want to get as much as you can in before you hit, one, the stomach acid, but even more importantly, the bile salts. What are bile salts? Emulsifier. Took the time to make this perfectly sized, perfectly controlled little lipid particle, and then I'm going to throw a bunch of bile salts at it and expect it to be the same? It's not going to be. So you need to design these real well to get fast absorption before you get to change by the bile. And of course we do glutathione this way. We do vitamin C with R-lipoic acid. Remember R-lipoic acid being that switch. And we do EDTA with R-lipoic acid. So I'd said that the basic glutathione upregulation will do mercury, cadmium, and arsenic. It will stop all the toxic manifestation of lead, but it won't get it out of the body. The EDTA is phenomenal for that, and the EDTA overlaps into the cadmium, the arsenic, allegedly the aluminum. I haven't seen as much of that coming out, and it goes into nickel as well.
So just going back to the phase 2, so we think all of those products are the ones that you're supporting phase 2 with?
Phase 2.
Question, please.
Phase 2. Okay, so Scott asked, what are we supporting phase 2? The clearway cofactors, that was what I got the warts off of my kid with. That's got the Haritaki, which has got phase 2 upregulation as well as biofilm busting. That formula also has nattokinase in it, which of course breaks the biofilms as well. So you've got Haritaki and nattokinase for biofilms, Haritaki along with the pine bark extract, and a couple other polyphenols in there for upregulating phase 2. It also has Gatucola and Bacopa for neurological detoxification and to stabilize the mind stream. Bacopa It was the only thing that really worked for ADD. Gatu Cola never really had that effect that Bacopa did. And when you're going through detox, there's a lot of destabilization of the mind stream, I like to say, like crazy making. And that's good for that. And then we also have so there's also our lipoic acid in there, and then we have our lipoic acid in the liposomes as well, with the vitamin C and with the EDTA. So you can either, we sell this sort of all bunched up into what we call our detox cube, which is sort of the deluxe. It's got the glutathione, it's got the C-lipoic, it's got the Clearway. You can have the EDTA as an add-on, it's got the IMD. But if you want to mix and match, there's different ways to get NRF2 upregulation just by having the lipoic acid in the C or in the EDTA. So there's a couple different ways that we hit phase 2 upregulation. But basically it's polyphenols and our lipoic acid. But now we know, as long as we've got the system working, we can go in there with pro-oxidants as well. So I told you all about IMD, and then phytogenomics, we talked about that. Talked about all that, and just to finish up on liposomes. Liposomes, there's a lot going on in liposomes now, and if they're done well, it's just awesome, because we're bringing in phosphatidylcholine, the very things that our cell membranes are made out of. There's our cell bilayer membrane made out of phospholipids. We bring phosphatidylcholine in, which is your most healing and most abundant phospholipid in the system. You're going to use the phospholipids in the liposome to rebuild your cellular and mitochondrial membranes at the same time that you're getting the nutraceutical that's coming with them. There's a fusion of the liposome with the membrane that makes the thing that you're bringing in there far more available to the cell. That's the beauty of the liposome. In fact, with the glutathione, they showed in cell cultures that liposomal glutathione was over 100 times more effective at raising intracellular glutathione than an aqueous glutathione like you do from an IV. So there's a lot that comes with liposomes, but they have to, again, be made well. You need the right size for absorption. You need high-quality phospholipids so that you can get the absorption correct. There's a lot of movement now on whether we're going to use soy phospholipids versus sunflower phospholipids. And if you're using the highest quality phosphatidylcholine, it really doesn't matter, except from a political perspective. The reason to shun soy is just to shun the soy industry. It's not because you're PC. I mean, all the, how many lipostabile IVs have you done before, THRiiiVE? A ton, and you're using high-grade PC from soy. That's not the problem. It's, it's more of a political problem. But just so everybody knows out here who's my client now, and you're wondering, why is my glutathione sunflower, but the C is still soy, the EDTA is still soy? It's a supply chain problem. I am like struggling daily to make sure I have enough of my materials to make these things. If I switch one more of my products over, the whole train's going to fall over. And I mean, that's how it is right now. And so bear with me.
Are they organic or non-GMO soy-based products?
Repeat the question.
She asked if they're non-GMO soy-based phospholipids. No, they're not. And I don't make them. This is a supply chain coming from Germany that's been making these things. How long has Lipoid been around, 80 years. These guys know what they're doing. They're making fantastic product, but they never saw the need to get organically non-GMO soy to do this. Now you'll see from some people they say made with non-GMO soy. I really doubt it, because it's not out there, and if it is out there, it costs a fortune, and it wouldn't be cheap like that product is. So they did testing, and none of the GMO proteins are soy is the Roundup ready proteins. None of those transfer. First, to get phospholipids, first you're taking an oil, so the proteins are held back in there. Then you're degumming it. You're taking part of that oil. Then you're purifying out the phosphatidylcholine. You're so far away from the components of the soy, they're not in there anymore. The phytoestrogens in the soy are not in there anymore. None of those things that you worry about soy for are a problem with these phosphatidylcholines. It is solely a political thing because they're not coming from, we're not just supporting these non-GMO soy farms wherever they are. We're taking out of that, and that's the only reason to switch over. It's not a biochemical reason.
They're outlawed in Europe anyway.
GMO is, yeah.
GMO soy is outlawed in Europe?
Well, all GMOs so far.
So far, yeah.
But it's, yeah, it's a very low increase.
Yeah. So I think they're taking from a lot of mixed sources to get their soy oil, and so they can't say it's GMO free. I know there is some out there, but it's incredibly expensive. And so I'm doing the best I can to sort of transition us over to good products, make sure that we're going to continue to have products as opposed to, oh, we're all sunflower, and then not being able to make everything.
You're doing very well.
Yes. Thank you.
Yes.
Thank you.
We're not done.
We're not done. We're keep going. Okay, so the point was we want to get really good phospholipids in there. We want to get good absorption. We want to get up to the brain. We've got a GABA with L-theanine in a sublingual that you feel very fast and very potently. Now, why would we want GABA L-theanine? So we just talked about so many things that happen with overexpression of the glutamate system, and we want to counter that by calming the system down. What we found is a lot of people, you go to detox them, you give them like one drop of a detox remedy, and the system goes all inflammatory, the brain goes all crazy, and that's overexpression of the glutamate system. But we found if we gave them GABA first, that would settle it down so we could give them a relevant dose of detox remedies or of antimicrobials for their Lyme. So that was real important. And we, GABA, people are usually taking grams of it to get any effect, but we do it in the right size liposome with the right lipids in there to go sublingually and get across the blood-brain barrier. You feel it very, very quickly. It's very effective in calming the system down. So there's a lot that's going to come. We've got methylation formulas coming. We've got a methyl B12 coming. We've got antimicrobials coming. And so hopefully we'll have a nice broad line for you to do all the tools that you need.
One of the things that happens in borreliosis is a splitting of the glycoproteins in the brain in order for the bug to produce the energy that it needs for itself. And a byproduct of that splitting of Borrelia of those glycoproteins in the brain is ammonia. And when you have ammonia excess and you take GABA, theoretically you can make more glutamate. So I didn't know if you've seen that before.
Certainly, I could say this about using GABA. When I first made this, I went down to Texas and I went to this woman's house, and she had some autism mothers come, and they brought a kid who had a severe anxiety reaction. He was freaking out because there was this dog there that he had a bad time with before, and he was just freaking out, wanted to leave. We gave him 4 shots of this, and he just settled right down and said, all right, I'm just going to go in the backyard. He went out and played with a kid he'd never met before. I mean, it was totally rad. The parents are crying. They became distributors. And so what did they do? They gave him that same dose 3 times a day, every day, as if that was going to keep everything normal. And by the end of it, it was an inverse reaction. It would wind him up. So you can, this is something you make. You can bleed some excess into it to bring the system down, but you can't just keep hammering after it, or if your body's set point wants to be crazy, like wound up, you can get a window of opportunity, but you can't force the whole system. So that was one example of where the GABA was, the body learned to turn it into glutamate to keep its set point.
I only wanted to mention, because we're all looking for better and better answers. Zeolite, which has been used in animal agriculture for years, totally takes a hog or chicken farm and stops the ammonia smell completely. And in fact, provably there's an increased lean body weight gain for the same amount of food. So somehow the nitrogen is getting used.
Yes.
And I throw that out because we worry a lot about ammonia as part of this picture. And we've wound up, because we do a lot of work with the pulsed electromagnetic field. And I can take people like Robert Rowan, who, because he lives 100% organic, 100% vegetarian, thought that he was above the rules. And so when he got his PEMF, he didn't wait until I got there to tell him the first day it usually don't go above 10 minutes. And so he treated himself for an hour. And the next day he couldn't walk and he could barely talk, and he didn't know if he was dying of flu or if he was just dying of detox. So we've learned that we need all the tricks that you're talking about. There are not enough tricks. I did, when I threw together our zeolite, we used the all-our-lipoid, a really good form of glutathione, and we added humic acid that makes zeolite do things it didn't do before. And we worked with NASA because there's only 2 legal zeolites that are legal in 40 countries. One is the one that they call natural cellular defense, Wyora. They're legal. They actually did their work. Everybody else is breaking the law, and they don't understand that they're not GRAS just because they buy a product. They have to do this separate research. And so because we wanted to go international for a very large multi-billion-dollar company that wanted to treat all of China, we spent a lot of time on this, and we did wind up recognizing that we can do pretty good stuff with this cation exchange story. And in fact, it does turn out that there is a negative charge to the zeolite when you get the right product. And of course, that product could fall into a poisoned area, and it could soak up all the arsenic, and it could be very poisonous. So you have to go through rigorous testing.
Yeah.
But in order to actually say that it does do something miracle, so it's important to throw it in our minds. We're interested in this ammonia story, and we're all looking for what is going to be the total answer, and there's no one answer. But I'm so glad you stressed the unique point about the all-lipoic acid, because people don't understand how important that's. It's not that much more expensive, but it's that much more beneficial. I sure appreciate the talk you're giving.
Thank you.
I just want to reiterate some of the things that Gary just said and keep it in simple form. There's a professor at the University of Cologne in Germany who wrote a number of papers on zeolite, and he gets a special zeolite from Cuba, which he's not examined, and that's supposed to be the right size and everything. And he shows that it works really like an extraction agent of ammonia from the blood. So it really.
I remember when I was a geochemist, and there was an old guy who just loved zeolite, Dr. Simpson. He's passed now, but they used it for gas absorption out of smokestacks, and ammonia was one of the things that it was excellent at sorbing, and so it would pull all this ammonia out. And then you're going to have a lot, you take that away, you're going to take a big stressor away from the system, and there's going to be a lot of cascades of benefit that come after you get rid of the toxic gas.
But it's important to know, it's really important to know that zeolite does give off aluminum in the gut, and Vyora is not innocent in that way. So it's
they've dissolved this stuff.
It's just important. It gives off aluminum in the gut, and so it should always be, at least in Klinghart's opinion, given together with an effective binder for aluminum. So it shouldn't. Well, chlorella, for example, binds it, and there's other silica, any silica compound will bind it. And so it's just also for you, it's really important to understand that. And the other thing that stops the ammonia production that is unfortunately not known in the US, even though it's plenty published, is the sugar galactose. Galactose completely stops the production of ammonia in the brain, and so you don't want to just extract it from the blood, because that's not really where the problem is. It's in the brain.
You want to stop the formation in the brain.
Formation of galactose.
That's why I use the galactose.
How do you dose galactose?
How do I dose it? Yeah, it's well, in an adult, it's a heaping teaspoon, 5 grams twice a day, and it's a 160-pound person, and you have to do the math on that one. And I know BioPure has the one that has been cross-examined and
yeah, that stuff's great.
stuff. So it's an important thing because there's a problem with pretty much everyone.
Yeah, yeah. But towards the binders, I mean, I really do want to get the right combination of all the different binders together. And we did start putting thiol groups on zeolite so that the zeolite would be able to do a lot of its own binding plus have the metal specificity as well. And so I'm trying to figure out the right blend to have, you know, the biotoxin binding, the solvent binding, the gas binding, and the metal binding, and the endotoxin binding all together in one product.
But that's interesting. If the zeolite gives off aluminum, do you really want liposomal zeolite?
Oh, no, nobody's talking about liposomal zeolite.
Oh, I thought you said something about that.
No. Dietrich may have mentioned liposomal silica. I didn't talk about liposomal zeolite. Yes, I did talk about thiolated zeolite, so it's got metal specificity. But a zeolite is something you want in the GI tract, not in the body. And it's also, you know, hundreds of times bigger than the liposomes. So
Robert, then Andre, then Gary.
Let's not forget that ammonia and ammonium ion exist in equilibrium in an aqueous solution.
Mm-hmm.
It's completely dependent on the pH, how it shifts. I really don't recall. I've worked with zeolite for 30 years as a water treatment substance. It's very common. It was the first water softening media for removing cations from water hardness.
Yeah.
Okay. They would charge it with sodium just like you do a water softener and then run water through it. This was the first work of Emmett Culligan in the 30s. Okay. Culligan man. So I really don't recall gaseous ammonia being removed by zeolite. It's possible. But it really acts on the ammonium ion. It's an exchange process.
Okay? And it's a cation exchange.
It's a cation, and it's nothing is better than this for removing ammonia and ammonium ion. But I am curious about. I've never settled in my own mind if zeolite it actually enters the bloodstream. Okay, this is like
any zeolite I would use would not enter the bloodstream because I'm going to have it at a particle size that is not going to enter the bloodstream. If they got to get it really, really, really small to enter the bloodstream. But like Wayora seems to dissolve it to ions, and so
I don't see. Once you dissolve it, you lose the.
It's not zeolite anymore. It's not a sorbent anymore.
And it's a brutal particle. I mean, it's like a guillotine.
Yeah.
And so it never made sense to me how this.
Yeah, I mean, even the stuff, I measured Wayne's sized his, and that's about as low as I'd want to go, and I think that's great. It was like 800 nanometers, but that's probably not going to absorb.
I'm also amazed, truly, I don't understand it, I want to understand it, how aluminum is liberated from this aluminosilicate.
Oh, it just dissolves with acid. But you just dissolve that with acid.
Yeah, but in the acid, like where is the acid? That's this level. Are we just talking like in the stomach? Is that what we're talking about?
In the stomach, you'll take some of it off. It's not like you're going to solubilize the whole particle. But you're going to pull some ions off.
Okay, interesting.
Let me say something quickly. That's not a matter of opinion. That's published. That aluminum is released from the stomach.
Yeah, it's got to be.
Small the amount in relation to what may be in average junk food, but if somebody pays actually take care of it.
Closer to your mouth.
Yeah.
So somebody actually is eating a clean diet.
It's not hard to do. I had a friend who did his whole PhD on aluminum released from soils from acid rain, and that's only pH 5.
Okay, Lee, then Gary, then Andre.
Yeah.
Lee, you cut in line. I'll forgive
you. Excuse me. I don't know if that study that Dietrich referred to was from healthy volunteers, but healthy volunteers usually have a pretty decent amount of hydrochloric acid in their stomach. But most of the chronically ill patients have very little hydrochloric acid in the stomach. Like Jonathan Reisman very rarely ever finds anybody with a pH below 5 in their stomach. And so it's drastically reduced acid production in the stomach of chronically.
That's almost none.
Yeah.
Okay, Gary?
Well, it's kind of fun when we talk about the idea that zeolite might capture gases because there's a lot of interest in this country making a hydrogen car. And they're very concerned about the hydrogen car getting hit and maybe the hydrogen being unsafe, so they chose zeolite as the safe way to keep the hydrogen in future cars.
Yeah.
And that kind of led to Wayne and I playing around and getting that minus 450 ORP stable that we'd never seen as stable. So we think there's something interesting about the zeolite, but in Croatia, they had a mechanical processor to try to get their zeolite down to a smaller size. And they wound up treating advanced pancreatic and colon cancer in humans. The dose is a teaspoon every hour of the day for 12 teaspoons a day. So it's kind of an interesting process, and they have lots of documentation, which I chose not to get involved in. They were hoping I'd help bring it to America. But it turned out that the zeolite we had uncovered because we were working with NASA, because NASA needs a good zeolite, because when you send a man to Mars, there's no water for him to drink. He gets to live on his urine and sweat for 2 years. So that man really wanted a good water filter, which has been pointed out, zeolite is the ultimate really good water filter. So we had a pretty good product, and I chose not to bring the zeolite in from Croatia. But they sell it in small jars, $500 a month, and they really have some great results. But
do you know what size?
Pardon me?
Do you know what size?
It's pretty big. It's not that small. You're trying to keep it in the gut. I chose 2 gut-related cancers. But it was interesting that they have lots of data that the dogs that get this as pet food, they don't get the cancers that they get otherwise.
Yeah, I saw the papers where they had the cancers on the dogs.
Yeah.
And they cured it. Those are even skin cancers, so on the outside, but curing it by cleaning up the inside.
Yeah.
Please cut it again. We love it.
Yeah, no. In the study that you were quoting, Gary, where they're using it for cancer patients, was that like a powdered zeolite or a liquid zeolite?
That's a powder. I have it sitting on the shelf at home. It's available. One of the FACT members is a board-certified anti-aging dermatologist who practices in Croatia. He sends me lots of stuff. Beautiful pictures of before and after of skin rejuvenation, lots of stuff. They have so much data that it takes more time for us to learn about. So I think the next person has this.
And see what doses. I mean, those are big doses. And there was I was into clay before I was a chemist, and it was the French did all this work on clay.
Yes.
Michael Abessera and those guys before him, and they would load on it. I remember I did all kinds of crazy stuff to myself with clay. Like, oh, my foot's falling off. I'm going to soak it in clay until it gets better. And people were like, you're going to the emergency ward tomorrow, and then tomorrow the foot would be like regrown. It was pretty impressive, But you got to use a lot, You really, you get bags of it.
Wait, Sarah has a question.
Bentonite clay was around a long time before we got into zeolite.
Zeolite's just super clay.
Thank you. That's a good term for it. Thank you.
That's all I was wondering.
Yeah.
What type of clay?
It was bentonites mostly. There was zeolites, but bentonites, montmorillonites were the best, in the original work. And like I said, I'd learned from my geochemist professor that clay, because of these long, thin structures that pulled the gases into them, and because they used them in smokestacks to purge take the gases out of there, and it was really just a super clay. It just had more extraordinary
air refresher.
Yeah, that's hence we just suck it down the tube.
Is clay bound to metals like zeolite is bound to aluminum as well? And do they?
No, it's part of the structure. It's silica and aluminum structure.
Right, I understand that. So is there metal in the clay structure?
Oh, is metal substituting
because
for?
I've had patients who've done these weird clay protocols and then done IV chelation on them, and I've got mercury levels of 500 on UTMs. And I was wondering if
it's got to be a downstream reaction. Clays won't have a lot of mercury in them.
Do they have other
They
metals?
do have other metals in them. Like you said, arsenic can get high in them. iron, but that's not as
Do
much
they
of
give
an
up
issue.
their metal? Because the whole zeolite people say, well, the
Oh, it's
aluminum
just
isn't
stuck
really
in
good
there.
enough,
Well,
but
it
in
depends.
an acid environment.
It all depends.
is.
If it's just sorbed onto it in the acid environment of the stomach, it'll desorb. If it's built into the structure, it is not going to very much, but you always going to get a tiny bit of dissolution. I don't worry about metals in the clay being a source to you.
Kind of like in cilantro, because it's such a high, it binds metal. So again, it can be contaminated easily, but it may not give up its.
That's
metal.
a different thing. Cilantro is not really a binder. Cilantro is a provoker of expression of detox enzymes. But like people are always like, why don't you put cilantro in your stuff? Everything that's in my system is stuff that I can document in some way that there's research about. We know what triggers it hits, and we know it works, but I can't fit it into my story, so I don't use it. But I know that it's triggering expression of enzymes that's making the mobilization happen. So I wouldn't call it a binder where I wouldn't worry about the metal in the cilantro. But I've never heard of cilantro having a big metals load. I don't think
because
that's...
people talk about it being a good potential metal binder, but you're saying there's no evidence of that.
No, and then they think it's hyper-accumulating metals. No, I don't think that that's true, and that's one of these things that irritates me is, well, because it does this, then it does that, then it does that. It's like, well, you're really not qualified to talk about that, so it's better you just shut up right now. But I didn't say that.
So you mentioned clay as not a good binder for metals.
Not the heavy metals. The heavy metals don't, even though they're technically cations, they don't really do cation exchange. Others, iron will do some cation exchange, calcium and magnesium, they do more of the cation exchange. And so it's not so much a binder of heavy metals. It might get a little bit of lead, that's a little bit more electrostatic, but it's not going to bind mercury. I mean, you put mercury in seawater, put zeolite in there, it's not going to bind to the zeolite. But it's going to do so many things at a GI level, including it's going to bind a lot of pesticides and herbicides. It's going to bind some of the mold toxins. So you got a bunch of mold toxins, almost all of them stick to carbon, but aflatoxin doesn't stick to carbon at all, but it sticks to clays beautifully, and zeolite, especially zeolite, beautifully. So it's going to take away a lot of the toxins that are holding the system down, allow the system to normalize itself and start working again. And then all the old French work on using the clay internally, it was doing something to normalize the epithelia. Like, I mean, I had really bad wounds that I pretty much had a big, wide open flesh on my foot. Fred, I don't want to tell you how I got it, but it was like, and I had this infection, like, dissolving my foot. That was basically, I was doing work outside, and I got this, this thing that was dissolving my foot, and then I tried to kill it with lye. I just like drove it in deeper, and soon it was just like a big open wound, and so I just slapped clay on there for days, and this is the one where my girlfriend was like, "You are going to the hospital tomorrow," and then I like peeled it open, it was all fresh skin. And it does have this regenerative effect. And so if it's going through the GI tract and doing this and fixing the epithelia, then it's going to restore the it's going to restore the effectiveness of the detox system, and you're going to move the mercury out. Like when you use the alginates and pectinates, if they're healing, reboot the detox system and move the mercury out. And then you see those two and you say, wow, pectinate's or citrus pectin's a metal binder or a mercury binder. No, it's not. But it's setting up the conditions at which your body can do what it was supposed to do.
That was a very important explanation. Thank you.
Yes.
That's
I was
excellent.
waiting for
Okay,
you
so
to mention
repeat that
pectin.
one.
You didn't mention pectin before.
I did. There was just too much going on.
Okay, well, we have a question from Anju, and we have an amen in the back from Gary Gordon. It's worth a repeat by Chris. Just break that chunk down.
Back to clays.
Just that last chunk.
Just the idea
pectins
that
and
just because you see that metal load going down
doesn't mean
that you have a chelator.
That you have a chelator. So modified citrus pectin is touted as a chelator. Zeolite is touted as a chelator. They're not chelators, but they restore the effective operation of the GI tract and thus the detoxification system, so that your natural chelation system, the glutathione system, turns back on and starts to work, so you see the mercury levels go down. That's why people then associate them together, even though that wasn't the right mechanism.
It's got somewhere to push it.
You heal the GI tract, you take away the inflammation in there that opens up the transport proteins that transport all the metals down there, and you get rid of everything again.
Okay, Keith. Suzette, one question.
Only one,
So-
Keith?
Yeah. So that explains when I took Wyora, and I always do high doses when I try things.
Yeah.
So the little bottle, you're supposed to take 10 drops 3 times a day. I took 30 drops 3 times a day.
Yeah.
And didn't feel any effect. Got to about the 6th or 7th day, ran out of the bottle. That night I got a headache. The next morning, my immunization shot blistered, just the shotted area, the scarring area.
Yeah.
Totally
you
blistered.
when you were little.
When I some kind of stimulation then that started to suck something out of
Kicked everything back up.
and
Pull it
now
on
the scar that was bigger, it's half the size, and it was red for like 6 months before it healed.
Yeah, so you reactivate the system, and you start sucking out stuff that, it's kind of like the housekeeping model. You know, you ignore something until you get to a state of hypervigilance, then you start cleaning up stuff that you had left in place for years.
That was like an analogy, Chris. I think that was kind of.
Did I use an analogy?
Was that an analogy? I missed it. He gave me grief about trying to dumb everything down.
I just, you know, there's times when we're ready for analogies, but if we go there before we're ready and then we lump analogy over analogy with a little metaphor and a little relation, and then you're just deep in it.
As long as it's like, you know, all 4 colors, it's good, Chris. Okay, Terry, come up, please. Terry Ranka. Autism will go too far.
So it's probably tempting for parents who have children who didn't do well on DMSA, which you've mentioned previously, to try something like CD, which you've mentioned previously. But are the children who didn't do well on DMSA susceptible to also not doing well on
You're
CD?
so smart, Terry.
Okay, repeat
Yes.
repeat your.
So she said, you go down the DMSA lane and you crash. So you think, well, let me go the opposite way and we'll go down the CD lane. And if you crash on DMSA, are you likely to crash on CD?
Chlorine dioxide.
Very, very, very likely. You know, this depends on dosing and such, but it's likely that most of that system that we're talking about was not in place, and that's why the DMSA did not work for you, like it did not work for me. And if you try a pro-oxidant, you're going after the same switch. It's probably not going to work that way either.
Robert, are you ever going to answer that question? After Robert. You can.
Which question?
I knew that was going to happen. Robert, come ask.
What question was I supposed to answer?
We'll get there. I'm going to let Audrey ask it.
Normally,
She doesn't even know what the question is.
Normally, when zeolites are used for any kind of chemical absorption or ion exchange, they are prepared by acid washing them. Okay?
Yes.
That's the real way laboratory zeolite is prepared for all these jobs. Another thing, I happen to have a study here on my computer that any of you, if you're interested, you can get it. Clinical evidence supporting the use of an activated clinoptilolite, which is one of the most common forms of zeolite, suspension as an agent to increase urinary excretion of heavy metals. Yeah, so
Hey, can I see that screen again? I didn't hear you.
there was clinical evidence supporting the use of an activated clinoptilolite suspension as an agent to increase urinary excretion of toxic metals. And one of the things they mentioned in here is that when they did an ICP-MS of the clinoptilolite itself, it was below detection levels for all the heavy metals.
Yeah. And in that study,
the most common kind.
in that study, they dosed pretty high doses of zeolite, and they saw metals in the urine come up and then go down. And I don't know exactly all the mechanisms at play there, but if you go back to that slide where you block the root out through the liver, you dump into the kidney, and then eventually the kidney starts working. If the zeolite is going to go through and clean up everything at a liver GI level, and then that starts working better, and if you take away inflammation from the kidney can release its load as well. And this may just have been where it's stopping a lot of inflammatory reactions and opening up the system. But it did go up and then come back down to where it was. So there is this release of a certain amount of metals that then slowed down. So I think, again, it goes back to this normalization of function in the GI tract cascading into normalization of other places. Because again, if you go back to the lipopolysaccharide on mercury, stopping the kidney from excreting mercury. So there was a dysregulation there, and perhaps the zeolite is releasing that lock that's going on there. I just don't know all the cascading reactions that are happening there.
When you order zeolite, the operative questions, this certainly should be, can you show me the test showing the amount of arsenic?
That's the biggest problem.
Arsenic is so ubiquitous in the Earth's crust, and that's where zeolite is just dug up like dirt by big steam shovels. You really have to watch that. I mean, I've had a lot of zeolite in my life passed, some from Cuba, through NSF, and some of it passed and some of it was horrible.
Yeah.
And it's almost like you can just get zeolite from a particular mine, the one which is considered one of the top mines in the world. And you know what? When you really find out what's going on, every shovelful is different than the other shovelful, or it's different in January than it is in March. So you really have to stay on top of it. But one of the questions I wanted to ask Lee is, what do you mean when you use the term liquid zeolite? I think that's really important because we glossed over it. But what do you believe it is? Do you believe it's just a suspension of, just a suspension of fine particles? Okay, good enough.
That's it.
I asked that question. I was just trying to get an idea about how much total milligrams of zeolite there was.
A teaspoon of sand. It looks like sand.
Yeah, it's a lot of zeolite.
12 teaspoons. Each
of powder.
works and weighs as much as sand.
Yeah.
I just wanted to say, this is more for the audience.
And look over here.
Some warning you when you look at studies like you just did. You give something and increases your urinary output of a toxic metal. I tried for 30 years now to find agents that actually decrease the urinary output of a toxin and increase it in the poop.
Yeah.
That's where we want to look.
Yeah.
So that's how we changed the whole way of detoxing metals, but finding desperate ways to stop the kidneys from being the resort. Because the kidney has a filtrating membrane surface of about 1 ping pong table.
Yeah.
The gut has a filtrating surface of 2 tennis courts.
Yeah.
And if you push a highly oxidative destructive agent through a surface of the body, you want to use a larger surface that dilutes it.
Well, it's also so slow to regenerate.
Exactly. The kidney, the nephrons are for life. The intestinal lining repairs every 3 days. And so looking at the studies that increase the urinary output of something is the wrong place to look, and so it's really important to know that's how actually got to the zeolite poop,
And that was,
looking for zeolites that are not absorbed, and that was my whole grudge with Viola. In the beginning, kind of marketed their product by showing the increased aluminum amount in the hair, and that really, really worried me. It really, really worried me, and so they, became quiet on that end. But and so we want to have we want to have a product, that Gary has and others have.
Go through the GI tract. I mean, that was what happened to me. I had 17 amalgams, and I just wanted to measure what happens when you take the MSA, and I would take the MSA, and measure my urine, and it would knock me out, and I'd feel horrible, and my urine levels barely came up, which means such a state in my kidney already that it all just dumped into the kidneys and stopped there and didn't get through. And I kept taking more and more, not having much more come out and getting really sick. And that was when I was at my sickest, is when I had the thought to go back to the GI tract and thus put the chelator through the GI tract and pull everything back over there. And all I want to do when I look at urine to blood ratios when we test, we don't want to see low urine to blood in organic. It should be at a certain ratio if everything's filtering right. I don't want to blow it above that ratio. I want to get it back to normal, but I want to, you know, move as much as I can through the GI tract. And that's what you'll see. You'll see you start clearing it out through the GI tract, and the kidney levels actually start coming up. And they should come up to a certain ratio between blood and organic mercury and urinary and organic mercury, and that means you're fixed. And there's only sometimes just clearing out the metals through the GI tract fixes that. Sometimes you need a couple other products, and the only two products I've ever seen work for that were Soluna's Renalin, the spagyric tincture, and Ayush Herbs Rentone. Rentone being the stronger of the two.
Can you spell the middle one? Spell them all.
They're both professional brands, so you got to get them through your doctor. One is coming from Germany. It's raised biodynamically raised herbs, raised in the Alps, compounded in Germany, and it's called the brand is called Soluna, like Sol, like sun moon, Soluna, and the remedy is called Renalin. And the doctors can get it from Innovative Medicine in New York. Tell them I sent you.
And we'll figure out a way for you to
They
get
have a
it too.
test kit for muscle testing.
What's that? And they have a test kit for muscle testing. They have. I use all their drainage remedies in the beginning. Detox. They have a kidney, lymph, and liver. Right? Remember, I said you need the liver is bitter herbs to drain the bile. The kidney stuff are diuretics to drain the kidney, and then the lymphatics are blood cleaners and lymph movers. But the Renalin is the one specific for the kidney, and it'll change that urine to blood ratio. Until you change that ratio, they're very hard to detox. Even when you're going through the GI tract with this, you are shaking the tree because you open up the cascade, and you do shake a lot of metal free, and it's going to distribute. Even though you're trying to go to the GI tract, some's going to hit the kidney. So if you start doing this and they have kidney pain, you got to back up and do more kidney drainage. The second product is from Ayush Herbs, A-Y-U-S-H. They're Ayurvedic herbs, organically grown in India. These are 7th generation Ayurvedic physicians. Import this into the U.S. They're up in Seattle. They're Bastyr guys. And they make a kidney formula called Rentone, R-E-N-T-O-N-E. And it's a wonderful, wonderful formula.
Is there a website?
We sell those at
Google will tell you where they are.
Home.
There you go. There you go. But that one's very good. And real sensitive people often have to start with the spagyric. Spagyric is in between a herbal and a homeopathic. It's energized herbals. And often they'll have to start with that and move to Rentone. People pretty distinctly test for one or the other or both, but often they won't test for Rentone, they will for Renalin. So you normalize that, everything will work well for you after that.
Let me go first for a second. Just to clarify, if anybody's listening and they're freaking out because they don't know how any of this stuff is spelled, Scott Forsgren is taking impeccable notes right now.
Scott will save the day.
He's saving the day. Now here's Terry Uranga. Sorry, I'm losing my voice.
So, Dr. Shade, are there foundational things that people will want to do first for the gut before shifting to move more out through the gut than through the urine?
Absolutely. It becomes its own course on its own is how do you normalize the gut. And at what point do you want to get to? I mean, when you have dental amalgam, the amalgam is the strongest influence on what's going on there. And a lot of people, well, let me just fix my body completely before I get the mercury out. Well, it's just not going to happen. You've got to dovetail the two, and you've got to get to a point at which you're happy to start detoxing, because often taking the metal out will fix the gut. But you have to do at least some basic work on knocking back the parasites and the excess microbial activity and the fungal activity, especially small intestine and gallbladder infection. The gallbladder stuff gets up into the gallbladder. You might be doing ozonated olive oil. There's a million different herbs that might work. That's something that's a lecture in itself, and I'm not the one really to give that lecture.
Oh, I want to know who in here has had the most experience in using colostrum. Yeah, who's had the most experience in here?
In using colostrum.
Using colostrum, yeah.
You know, I remember meeting a guy who was incredibly mercury toxic and everything toxic, and he was close to the Amish farmers, and he went and got the fresh, not dried colostrum, the fresh colostrum, and he would buy it from them by the gallon and drink just that, and he fixed everything.
Are you asking because it's a good gut healer or what?
Gut healer,
right.
Getting rid of the inflammation, helping the gut get to a place where it can actually handle the toxic load.
Yeah, Sovereign Labs makes a nice liposomal colostrum. Yeah, it's
You're pissing people off now, I'm just telling you.
So they're asking about colostrum and his ability to take away inflammation in the GI tract, regenerate the lining of the GI tract, and set you up and change the immune system in the GI tract. I mean, that's one of the biggest things I found with getting rid of dysbiosis, that often it was immune. TH1 things like astragalus, which turn up your natural killer cells and turn up your interferon and turn down your TH2 response, will then fix your GI tract and enable you to control what grows in there. And so the colostrum would do that.
Yeah. With the colostrum, another big piece is why we breastfeed, right, is that it allows us to populate the good bacteria, and our kids have this imbalance. And so one way to populate good bacteria back into their gut is by using colostrum. And some of these protocols with these new products that are like Bravo probiotics, they want you to ferment them in dairy, so you kind of get that kind of effect as well. And using those types of probiotics with colostrum is sometimes pretty helpful.
Sarah's on that mic just for you out there.
I just wanted to say just a few words of getting the patient ready for detox. So most of the patients' whole metabolic system and digestive system and nervous system is shut down from the metals. And therefore, very often the first step is that you have to attack the metals. So there is sort of like a myth that you need to get the system ready to get the metals out, but if the metals are causing the shutdown...
That's what I was saying, you know, you got to choose a point at which you're going to go after the metals.
Yeah, and so we have a few ground rules. One is the kidneys seem to be okay. There's a new marker, relatively new, it's the cystatin C. It's a more sensitive marker for kidney function.
What is it?
Cystatin C.
Yeah,
cystatin. It's like, nystatin, it's just a C at the end, at the beginning.
Oh, like cystatin.
Cystatin C.
That's used
better than creatinine clearance.
Outside the US since 10 years or so, but it just came on the market here since a year or two. It's a wonderful, simple marker. It's related to creatinine, but it's just... And so we do a pH thing, the saliva pH needs to be more alkaline than the urine pH. That shows that the kidneys are able to actually bind things to acids and detox.
Yeah.
So we have that as a basic thing, and then the patient has to poop. No matter what you do, you can't start the detox unless the patient poops. And if that's artificially induced with a colonic every day, fine. But they have to poop. Without pooping, the whole detox is shunted to the kidney, and you destroy the kidney. And so these are the only two rules that we have in my office. We're doing very well since 4 years. We've never killed a patient doing this.
Yeah, and really, I think the only thing that I've seen that's helped as a pre-starter is a little GI killing. Even with the artemisinin, we use 2 bottles of liposomal artemisinin and then go into it. Just knock that back a little bit, because if it's anything things.
I'm so glad we talked about protecting the kidney. because the doctors who haven't learned yet the whole lead story need to pay attention that New England Journal of Medicine published a paper that if the patient receives 1 IV of calcium EDTA a month, they never wind up on dialysis, which would put billions of dollars back into the American economy.
Yeah.
And I'm so glad that THRiiiVE Klinghardt is mentioning the new test, because we have time for people to be aware that their kidneys are stressed. I have had to particularly fight with my chelation doctors because they choose to ignore the 507 published papers from industrial doctors that work with lead workers in lead batteries in the plant. The only way they can continue to report to work is to take their EDTA tablets. And oral EDTA happens to be only $2 more expensive than oral vitamin C. So to fight me on taking oral EDTA, you're going to have to fight with the cost of dialysis. And you're going to have to realize that the fact that as soon as you see any decline in kidney function in people who are not working with lead, even just diabetics, if you give them some EDTA, and if they don't want to do it by vein, it's documented with 507 papers on my website that provably EDTA by mouth is able to do a provoked specimen and show actually how much lead comes out. And so it's interesting when we plug lead in, because lead makes the mercury so much more toxic. And it's interesting when you look at the big picture. I take every day my zeolite and my vitamin C and my fiber and my maca and every green that you can buy and stabilized rice bran, because I assume everything I eat is poison. But in addition to that, we have this small problem that Mother Nature decided that the mom's life is more important than the baby. So the baby is a toxic dump site, and documented that the bones of every child at birth averages 1,000 to 2,000 times more lead than any bones on Earth just before the industrial age 700 years ago. So we're paying a little price for having our convenient cars and turning on the electricity and burning coal. But it's interesting that only Harvard has done the studies to show that blood lead is nothing as near as important as bone lead. And bone lead is not released by IV chelation, so it's only released as your bones remodel. So you've got 15 years to work on the problem. And each day, if you actually had lead-free air, a lead-free house, lead-free food, your bones are still carefully making sure we poison you a little bit every day. So I want everybody to understand that, to me, detox is a lifetime program.
Yeah.
Yes. Can I want to say something there? I'm trying to multitask here, so it's killing me. Okay, so now we're going to answer that question.
You're going to answer what question?
Exactly. But number one, with the detox, why wouldn't the IV EDTA or oral EDTA be the beginning of a solution?
Well, if they have nothing to do with, they've got to have something to do with each other. You just say, well, he talked about the kidneys, she talked about the kidneys, they must go together. That's kind of reckless.
But if you're heading for dialysis, your kidneys
that has nothing to do with detox. He's talking about long-term damage of the nephrons from lead accumulation. Lead, cadmium, and mercury are the ones that wreck the kidneys.
Okay.
And there's great data of creatinine clearance coming up after EDTA chelation. Now, the one slide I forgot to put in there was NRF2 again. When NRF2 is down, the kidneys are incredibly susceptible to oxidative damage. That whole mechanism there is required to repair. And when you get into the inflammation, all those metal transport proteins in the kidneys. And so when you see no metal coming out in the kidneys, in the urine, you're really at a bad place in the kidneys, and you don't want to move more metal until you get them, until you can get the kidneys functioning right, and that's having NRF2 glutathione processes in there working well. So that kind of defense, NRF2 upregulators while you're doing EDTA chelation, I think is what you want to do, because there's inevitably going to be some of the people that you try to move that lead out and you stir up even more of a fire. And so I think if you're That's like in our EDTA, we have our lipoic acid in there too. So we've got an NRF2 upregulator. At the same time, we have something that'll pull the metals out. So if we do everything right, we're going to resuscitate the kidneys. But this is long-term damage from metal accumulation. PTOX, as you're calling this incessant urination, I don't think it has anything to do with lead. I think that has, you're screwing something up. Oh, I'm sorry. You're challenging
it's
something.
Okay. Screwing up is okay.
You're challenging your patient with, endless amounts of whatever you're trying to detox them with.
So you got a patient. What do you do? Detox.
What do? You stop trying to detox them and you support the kidneys. So pull back from whatever you're taking. If it's chlorine dioxide that's doing that. Now, if I would go, I would go and try to fix the liver-GI connection. So there was one mom came to me, and her son had to wear diapers after doing too much of one chelation agent. And it set him in so he was hyperammonia and peeing all the time. And so it took me a while of ruminating on it, so I'm like, oh wait, it's that liver-GI connection. I told her to do methylation agents and liver cleaning agents, and it immediately slowed that down. So get things to actually, you probably put too much oxidative stress into the liver and shut down the gallbladder drainage of the liver, shut down the liver-GI connection. You're shunting everything over to the kidney, and something's going out of control. So stop, whatever you did to get there, stop that, and try to reopen liver, gallbladder, small intestine.
What about liver, gallbladder?
And then there are some supports, I forget all the ones that are like maca, some of the ones for ammonia. You got to turn down that ammonia production. TMG.
But what about a liver, gallbladder flush? And just quickly, in your opinion, we'll go around the room right now.
Well, I think, a kid who's under a lot of stress is probably not ready for a liver gallbladder flush, and a lighter way to go about it is the bitter herbs to drain the gallbladder. Do liver gallbladder GI support. You think everybody's ready to go right for a gallbladder flush?
The long-term damage, how long is it going to take the bitter herbs to work, and are they going to work sufficiently enough to get the gallbladder moving? If you dump it in 4 or 5 hours, you dump all this trash out, most of it's going to go down the GI tract and out because it's being pushed out by other processes, like the clean as a whistle and Epsom salts, which is killing a lot of bad bacteria that clogged it up. So if you push all that out of the system, then you freed up the liver, it dumps a bunch of crap out.
So
now it's functioning and putting less symptoms.
Keith is saying it's going to take me too long to free things up biochemically. We should go to this physical reflex of stimulating the gallbladder flush, getting all of the obstruction out of there, and making the whole thing work very quickly.
Let me intervene here. So,
please.
So when the kidneys are in trouble, one of the key problems is the enterohepatic recirculation of biotoxins. So when you do gallbladder flush, that's absolutely safe under one condition, if you have enough binders on board. And so the whole kidney thing, I actually conducted a study, one of the very few actually I did my whole life, that was with the University of Tübingen. That was in the early chlorella days. We examined the poop and the urine and the blood every day of 30 volunteers, and half the group we gave chlorella, small amounts in those days, and half the group we didn't. And the interesting thing was, in the chlorella group, the urine excretion of mercury completely disappeared in the chlorella group. And however, the chlorella group had a 10-fold increase in poop excretion of mercury compared to the control group. So control group kept peeing out small amounts of mercury every day, and had very little in the poop. And the chlorella group pooped out huge amounts.
Everything.
Nothing. That's where I learned about the binders. And so, however, the best binders we have today is the zeolite, and is your stuff, the IMD. IMD is fantastic because it doesn't get absorbed, so it doesn't stir up more detox. What we found is like when you do antiparasitic remedies, you know, I showed in the last summit, I showed the evidence in the literature. Parasites carry up to 400 times the concentration of metals in the body that the body of the host has. You know, so parasites actually bind metals for you. They're actually extracting metals out of your system. So when you kill the parasites, you get a huge release of metals. Many people get into near kidney failure when you do too strong antiparasitic stuff.
Okay.
And so that's why have become a big promoter of doing the rectal route first of antiparasitic treatment. You want to get as much whole parasite out of the gut.
Because when you kill it, you're gonna get inflammatory response from the dead organism as well as the metals.
Yeah, yeah. And so when somebody is in kidney trouble, we just give binders and the direct kidney support agents, you know, sort of follow up until the kidneys are back up to snuff. And then, you know, and then the gallbladder flush is fantastic to save the kidneys, but only after bringing the binders on board, which only takes 24 hours, you know, to have binders on board, and then you can do the gallbladder flush. So that's the only.
And I'd use a broad spectrum of them.
And we use Keith's stuff. And Keith has, I think, developed the best protocols on the gallbladder flushes. So I've seen.
Just to mention, homeopathy still fits in this world. I'm the president of the Arizona Homeopathic Integrated Medicine Association, and Bruce Shelton published a study in Explore, where he took a product from Heal called molybdenum. Then Heal is now moved out of the but he definitely showed enhanced excretion on IV chelation EDTA patients when you added homeopathics. So let's always remember, homeopathics can be very gentle, depending obviously on the X and the concentration you choose. But it's always a missing link that we may all be neglecting.
One other point about the liver gallbladder cleansing process is that we shouldn't forget is the emotional component. Because patients that have a lot of unresolved anger, frustration, will tend to hold on to toxins, much more so in their liver and the gallbladder and their bile ducts. But if you can get them to release the anger and frustration, then they start releasing that. You see that energetically, and I've seen it in a few collections of stool specimens and urine specimens as well. So if you're talking about a child, how do you get the child to release the anger? There's a challenge, especially an autistic child without speech. What I found is that you can actually do an emotional session.
Hold it by the base instead of the bottom. Hold it by the top. Yeah, there. The connection's making noise, so hold it there.
Yeah.
Like every second hand. Look at that. Perfect.
Okay. So what we found, if you have a child that's very young and without speech, that you do an emotional release session on the mom. So you get her to go out by herself and do some shouting and visualizing and raging and eventually forgiving. And oftentimes, if she'll go to the bedside of the child at night, right after she does that, and tell the child the story of what she did after the child falls asleep, the next day the child will wake up with less liver stress energetically. So, and if it doesn't work to do it with the mom, you do it with the dad too.
Back to Chris.
That's great.
Any final words of wisdom for this particular 3-hour session?
Oh, I think we've had enough for this session. I think it's time to eat. And
so detox. Back off everything that got you there in the first place.
Yes.
Make sure you're pooping, because the fact that you're not pooping is probably part of the reason why you got detox.
But it's back off everything you did. Go back to the GI tract and the liver.
And we've got all kinds of stuff that we'll put in the notes for dealing with the kidney, the liver, and all that. Okay, anything else? Thank you, Dr. Christopher Shade at QuicksilverScientific.com. Yeah, baby. Woo! You know what? Here's the deal. I'm going to leave this going. So why not? No? Okay, fine. I'll stop it. Bye, everybody. You got booted. Oh, I don't know. I'd